Good morning, everyone. Welcome to today's session on with the CCM Summer School. I think we'll just give it a couple more minutes for some people to join. Then we'll start maybe at 9:02. Okay, it looks like everyone has joined. So we'll probably begin with the session. And I'll be sort of just moderating the session today and introducing our speaker. Yeah, without further ado, I am it is my pleasure to introduce our speaker today, Professor Frank Cutter. Professor Hutter is a professor for machine learning at the University of Freiburg, as well as a Hector Endowed Fellow at Ellis Institute in Tübingen and the head of the Ellis unit in Freiburg. He is most well known for his many contributions to AutoML, including AutoWeka, AutoSKLearn, many of the benchmarks that we use, and also for founding the AutoML conference. We're excited to have him talk about his latest research into foundation models for tabular data, which yields exceptional performance as well as efficiency in low data regions. Frank, when you're ready.
Thank you very much for the invitation and for the nice introduction. Yeah, it's my pleasure to talk about small data predictions for tabular data with a new foundation model called TabPFN. So, so the premise is that machine learning for tabular data, that's what the talk is going to be about. And tabular data is extremely widely used. So think Excel tables, think databases, there's tables everywhere. So each row here is going to be a data point and each column is a feature. And then you have a special row, a special column that is blocked by the Zoom window for me right now. It's here, that you want to predict. And you have historical data, and then you have new data where this information is missing. So in general, it looks like this. We have some training features, some response we want to predict, or that we have actually for this historical data. Then we have some new data where we have these features, and we want to predict this response variable. And so we want to learn a functional mapping from this X to the Y based on the training data. And there's numerous examples. This is really ubiquitous. For example, in credit scoring, you might have features, could be the income of a person, credit history, credit amount, and you want to predict, will the credit be paid back? But more importantly, for this school here, in medical data, you might have as features the symptoms and the measurements, and you want to predict, well, what is actually the disease? And there is thousands and thousands of different types of problems where you have these types of inputs, and you could have, I don't know, protein markers in your blood, or you could have images. Well, you could have tabular features of images that sort of doctors have historically extracted, and then you predict something from it. So this tabular data is really super widely spread. Yet the machine learning methods for it are not what you might expect from what actually works well for other types of data. So neural networks in particular have really led to huge breakthroughs for unstructured data, where you have text, where you have images, etc. But if you have this sort of structured tabular data, then actually sort of the historical methods were linear models or SVMs, maybe random forests. And what's really being used these days is gradient boosting. We heard about AutoSKLearn briefly in the intro. There's AutoGluon. these types of methods work well, and deep learning has not taken over for tabular data. And that is partly because, well, neural networks excel for large amounts of data, but they're actually slow to train. So they don't really work well for this quick workflow that the data scientist wants to have, like load in some data, run a model quickly and see how it's working, etc. But they're actually quite slow. And more importantly, they're prone to overfitting when you have small data sets. And small data sets is what we actually have more often than one might think in this era of big data. We rather have many data sets that are small. So especially in scientific data, in biology, medicine, climate research, etc., you might have a rare disease, and then you have like 1,000 data points, and you're not going to get more than 1,000 data points. For some of my medical colleagues, 1,000 data points is a huge amount of data. So they might say, well, I have 10 data points. So even with 10 data points, we would like to do something meaningful, and deep learning would just stand no chance whatsoever. But then, sort of by its very definition, we just have a lot of these data sets, and this question comes up all over again. And as we saw before, there's credit scoring, there's medical data. The features that you have in each of these tabular data sets, they have nothing in common. It's not like in text where, well, this is an English text and there's another English text, and you can sort of just transfer some information from A to B. Or in images, it's sort of just all pixels. Here, it's sort of the features in this data set have nothing to do with the features in this data set. So it's much harder to actually build this sort of transfer across all these many data sets. And that is the core reason why tabular data is really, for the longest time, hasn't had its foundation model that vision and text and speech and so on have had for a long time. All right, sneak peek about what I'm going to talk about. So we meta-learned a very strong machine learning algorithm for small data. And it's going to feel very much like GPT because it's executed as a single forward pass of a transformer. But it nevertheless actually has state-of-the-art performance, doing better than all these classical algorithms for small data sets. And I'd like to emphasize that it's a very different approach to algorithm design, because we did not code this algorithm manually. The algorithm that will be executed is actually learned across many different data sets. And all that we did basically is to say, these are the types of data sets that it should work well for. And then we sort of have a machine learning algorithm like this one that builds another machine learning algorithm. So we have it's basically just standard in context learning, if you're familiar with that term, that is executed in a forward pass, just like in GPT. And that is learned by simply, the standard supervised machine learning loop that we all know from GPT, just the standard transformer. And so this algorithm that is being learned, that lives in the weights of a transformer, and the transformer in its forward pass is executing a machine learning algorithm, just like you would execute XGBoost or something like that. This is executed in the forward pass of a transformer, and this transformer is just learned from data to do well on the types of data that you specify. So it really turns the system on its head. Now, in order to get better performance, it actually really helps if you know more about your data. I think in a few years, data scientists shouldn't need to know anymore what an XGBoost is or what a hyperparameter is. They should know about their data. They should know that there's data drift, data shift, or concept drift, concept shift. They should know what classification is, what prediction is. But sort of in order to drive a car, we don't learn to become a car mechanic. We take a driver's license. We learn to drive. And likewise, with machine learning and also machine learning for tabular data becoming more mature, I think we should really move towards this data-centric approach where people learn to use the knowledge that they have about their data. And here in particular, you can just specify the types of data sets that it should work well for. Push a button and go learn an algorithm that works well for this particular data. So that's sort of the medium long-term vision. All right. With that as a sneak peek, let me get into the outline. First, I'm going to talk about prior fitted networks, which is sort of the algorithmic foundation. There we're going to see that transformers can perform approximate Bayesian inference, Bayesian prediction, so computing a posterior for a given prior. And this prior in the second part will actually be a prior over what could tabular data sets look like. And then we actually see some data of a real tabular data set. And by turning the crank, we get a Bayesian posterior over our prior, what the missing Y values might be of this new data set that we're getting. And this is TAPFN is a work we have published in 2022. And since then, we've been building and have a new version that I'm going to give some results about. This is yet unpublished, but under review. And I'm also going to show a quick demo, time permitting. All right, let's dive right in. So prior fitted networks. PFNs, they can predict the posterior predictive distribution for any prior that we can sample from. And they're a very simple method in contrast to MCMC, Markov chain Monte Carlo, or variational inference. There's nothing, no complex math involved. All that you need to do is supervised learning on data that you sampled from your prior. And that enables this very different approach to algorithm design. So PFNs are a GPT-like model for probabilistic inference. So you could cast this as a next word prediction. So you have X1, the features, Y1, the response variable for this first data point, up to Xn, Yn, and then we have Xn plus 1, the next data point that we're querying for, and then question mark. And of course, then you're supposed to fill in Yn plus 1. To be a bit more precise, it's not just a GPT-like sequence. Actually, we have a data set, so we don't care about the order. We could also put this data point here first and this here last. And we want to get the same prediction. So really, this is a set. And then we get prediction out. And even more precisely, we don't just want to get a point prediction, but we want to get a probability distribution. And that probability distribution should approximate the Bayesian posterior. So on a very high level, this is what we want. We want to feed this into a PFN and get out the predictive distribution. And this is going to be a transformer with parameters theta, and that single forward pass should then approximate this distribution. And we optimize these theta to minimize the average cross entropy loss across many different data sets in order to just directly approximate this predictive distribution. And which data sets? That's a tricky part. There aren't a whole lot of tabular data sets of high quality available. People have sort of parsed the internet and just throughout the internet gotten all types of tables from the internet, but these tables don't necessarily give rise to tabular prediction tasks like regression and classification. Yes, you can generate them, but they might not actually be what you want. So rather than relying on this also large data that GPT also did, we actually went to work purely on synthetically generated data. And that helps us to be really in control of what we want the model to do. We can specify exactly this is a prior, and we're going to build a Bayesian posterior for this prior. If we use all the data from the internet, well, you could get garbage in, garbage out. It sort of depends very much on then how high quality is your data. And we bypass that problem by just working purely with synthetically generated training data. We also bypass the problem of overfitting because we simply just never see real data for training. So there's no risk of having seen all the data sets that we might later evaluate on or evaluate in subsets and so on. Right, how do we train it? This is really extremely standard. It's just a standard transformer architecture. The only thing that we do is drop the positional encoding, and yeah, and we do drop the positional encoding because the standard transformer, well, that actually is precisely is the sequence, and we want to get to the set. And the only thing that makes a transformer aware of the position in the sequence is the position encoding. And if you drop that, self-attention is actually set-based. And so we actually simplify the transformer for us because we only need a set. All right, that's a very high-level intuition. From a Bayesian perspective, so this approximates Bayesian predictions. If you work on Gaussian processes, or if you know what these are, then you're very familiar with these pictures. If not, I'll explain it. So here we have the y value, and here we have the x value. This is one-dimensional. Of course, I can only plot in 1D, but you can think of this as some high-dimensional surface. And this is the y value you want to predict. And there's many possible functions. And a prior over function specifies the types of functions we expect. And this here is actually from a Gaussian process. It's a smooth kernel that gives rise to these very nice smooth functions that are infinitely differentiable and so on. And this is parameterized by some latents, namely the kernel hyperparameters. And then when we actually observe some data points, so we observe for this feature values, we get this target, we get this one, this one, this one, and this one. Then this constrains the space of functions that is compatible with this type of data. And we can build a posterior over functions that is compatible with these observations. And so that's you would build a posterior over the latent factors that govern these functions. And actually doing this is unfortunately intractable to compute in almost all settings except for Gaussian processes. Gaussian processes are just this one exception. What we do is we actually, so then you typically use this posterior in order to compute the posterior predictive distribution by using the posterior over the latents and multiplying it with the probability of observing the y's given the new x and these latents. And you integrate out over all the possible latents. So usually you would do this with MCMC, Markov chain Monte Carlo, then you would get point estimates for these t's, and then this here becomes a sum over these t's. Or you do it with variational inference, where you get a nice Gaussian predictive posterior here. Then this is a Gaussian, this is typically a Gaussian. Gaussian times a Gaussian is a Gaussian, integrate out is still a Gaussian. So these are typically the methods used in order to compute posterior predictive distributions. We completely bypass this step here. We just directly go here and only solve this problem for a given x. What is a predictive distribution? And while MCMC and variational inference are tricky sometimes, this here is just going to be a forward pass. Let me visualize it a bit more. So what we need is samples from the prior. So just like here, these features, these functions are plotted. So basically, we sample a function, and we sample another function, and another function, and we do this sort of 10 million times. And then for each of these functions, we sample some training data points and some test data points. We do this for each of the functions. Doesn't need to be the same number of training and test data points. And yeah, so we do this often. We can do this as often as we want because this is purely synthetically generated data. It takes seconds. And then you learn to map from the blue points to the orange point. So given the x value, what is the y value of the orange point? That's what you learn. So the blue points will be inputted to a model, and it's and the x value of the orange point, and then the y value, distribution over the y values is supposed to be outputted. And once you've learned that for 10 million data sets, or data, like a data set is like a function here, so for 10 million functions here, then you get a new function and samples from this function. So these blue points and the question, and you feed that to your PFN, and you just learn to, well, predict the probability distribution. And so with this, we have just meta learned to approximate Bayesian inference purely by supervised learning, purely by this PFN mapping from the blue points to the orange points with a standard transformer. And all we needed to be able to do is to sample from our prior. And this works. So this actually in theory is guaranteed to predict the true posterior arbitrarily closely. So if we do this approximation here perfectly, and we sample infinitely many samples from our prior, then our posterior predictions will be perfect, exact. And so here we have sampled, we've trained based on 1 million data points. Of course, a transformer doesn't get exactly to perfect predictions, but you see very small differences between the blue curve that is actually our prediction and the green curve that is, in this simple case of the Gaussian process, you can predict the posterior in closed form. And so here we get something extremely similar. And if we train more, then it's going to get even better. With a bit more math, you can visualize this as follows. So you sample prior data sets from a prior, these data sets here. Then you train the PFN to minimize the negative log predictive error. And you get out your PFN with these optimal parameters, and then your PFN with these optimal predicted parameters will actually predict the true posterior. All right. The model we use for this, I mentioned before, is just a permutation invariant transformer. One thing to note is we do not have any attention between the test data points. So all we can do is predict for a single data point at a time. We do not model correlations between the outputs. So with a Gaussian process, you could get the joint probability distribution of x4 and x5 given this data. With PFNs, you cannot do that. You can draw samples from it, joint samples as well. You could draw the y4 given the x4 from this distribution, throw it into your data set, then draw the y5 for x5, and do this 100,000 times, and you would get a joint distribution over x4, x5. But it's slow, and that's not what's done in the model typically. All right, and the other thing that we do in order to make this possible, in order to just have our output distribution actually predict a full predictive distribution that's not necessarily a Gaussian or anything, is we actually do regression as classification. So we bin our output space, and we do that actually by samples from the prior. So we have 10 million samples from the prior, we push them forward, and then we take the sort of the 10,000 lowest prediction, put them in the left bin, the next 10,000 into the next bin, etc. So these bins here are of varying size, and they're adaptive to the data. And if we have a lot of data here, then the bins are going to be very small there, so that we're very accurate where there's a lot of data. And then the leftmost bin and the rightmost bin, we make these sort of half Gaussians in order to not get zero probability for data that is sort of outside the bin. All right, we can also approximate more complex GPs. Works much better than the previous attentive neural processes. And here's also an evaluation against MCMC and variational inference. Well, actually, that's going to be on the next slide. Here's just how, just visualizing here. So here's a true GP. This is the true exact posterior that we want to compute. And here we see that longer training, so that's training based on more sample data sets from the prior. This is trained on 500,000 data sets, 1 million data sets, 2 million data sets, 4 million data sets. So it keeps getting better and closer to the true Gaussian process. And also the number of data points. Well, that's the number of data points you have seen, the number of the blue points. If you've seen a lot of blue points, then you get more and more certain about the function. The true posterior GP does, and so does a PFN. And here we could scale this to about 4,000 data points. After that, the transformer might be too slow. Why? Because of standard transformers are quadratic in their input size. but we all know that the context window of GPT and Claude and so on has been expanded a lot, and all of these tricks would actually directly carry over to this year. All right, here's the evaluation against MCMC and variation inference that I spoke about. So here we have the negative log likelihood that we want to minimize, and the PFN with 4 million data sets actually based on 10, 20, 30 data points is actually much better than MLE, maximum likelihood estimation, better than the NUTS. So there's no U-turn sampler. That's a pretty strong MCMC algorithm, and it has hyperparameters, and we tried different hyperparameters, and just none of them work as good as a PFN. We also looked at Bayesian neural network approximation with tiny Bayesian neural networks with like 3 features, 2 layers, hidden dimensionality of 5, because then you can actually do MCMC and variational inference, and they do get to the same negative log likelihood, but they take sort of 10,000 times more time than a forward pass of the PFN. And already with the slightly larger one with 8 features, we see that they don't quite get there. So this is really something that you can do Bayesian posterior inference even for small Bayesian neural networks. And so what we would have in these Bayesian neural networks, you would have weights over the neural networks. You would have typically little Gaussians at each of the individual for each of the individual weights, and then the posterior predictive distribution is the Bayesian neural net. And so there, as I just showed, you could be 10,000 times faster than MCMC, but you could also do other things like having a prior over not just the weights of a neural network, but also over what is the architecture of the right neural network for your data, and then the posterior predictive distribution would sort of be a Bayesian neural architecture search, which wouldn't really be possible with MCMC. You could do sort of reversible jump MCMC, when it would be horribly complex and would almost certainly not work very well. All right, and we don't give you the posterior over which is the right architecture, but we give you the Bayesian integral over the right architectures evaluated for your data. So what would the right architectures predict in terms of the Y values? Because we bypass the step of getting posterior over the latents and directly go to the prediction of the Ys. All right, so this was PFNs, an original paper from 2022. There's a lot of follow-ups by now. This slide is very incomplete. It's sort of old. There's follow-ups in tabular data that I'll talk about, for time series data, for Bayesian optimization that I briefly mentioned on the next slide, for medical images, for genetics, and so that there's more than 100 follow-ups by now. Bayesian optimization, I will briefly mention. So Bayesian optimization is something that we do a lot, and it's based on Gaussian processes, and Gaussian processes are great for being cute in terms of the math, and the math just works out and is beautiful, but they are also somewhat limited in the flexibility. So for example, if your noise is heteroscedastic for different parts of the input, you have different types of noise, or you have change points in your data, then Gaussian processes are not great, and so you can replace a Gaussian process with this PFN in context learning, and actually that gives better performance, as we showed in this paper here. You can also use it for learning curve extrapolation. So if you have sort of an initial learning curve of a machine learning algorithm and you want to know where will this go, then you can also use it for this. Actually, the first master thesis that happened in my lab, Tobias Domhan in 2005, he actually used MCMC in order to do stochastic learning curve extrapolation. And we took his MCMC method and ran it, and our new thing, and we're just 10,000 times faster now by just pushing it through a transformer. All right, you can also use it again for more complex Bayesian optimization, freesaw Bayesian optimization, where you sort of have many partial learning curves like this, and you want to predict where will they all go, and then run the most promising one for another epoch. I won't go into too much detail here, just saying that this also then yields new state-of-the-art performance for pre-sub Bayesian optimization. That was an ICML paper this year. All right, so I encourage you to use PFNs yourself. All you need is a prior you can sample from with a scalar target to be predicted. The optimal setup is if you can just sample a latent and then draw samples based on the latent. And yeah, we have a collab that you can walk through, github.com/automl. That's our repo, and then /pfns. That's sort of a self-explanatory collab to step through. All right, then next up, I would talk about how does this all matter for tabular data. I'm going to use these PFNs and just going to have a prior that is based on tabular data sets. So for tabular data, you have, again, the setting. You have X train, Y train, and X test, and you want to predict this question mark. So the Y test this here, you push this into TabPFN, it's parameterized by theta, that outputs some predictive distribution, which doesn't have to be a normal distribution. And then you look at the negative log likelihood of your test data under this predictive distribution and optimize this with SGD based across millions of different samples of these data sets. So not millions of rows or something, but millions of just different types of matrices here. And once you've learned to do it for millions of these types, then you get a new one and you push it through TabPFN, and it also makes a prediction. All right. So what is a prior for tabular data? There we thought and looked at the literature and so on, and we thought, okay, well, let's use some principles from causality, because the likely data in the wild, there's actually some causal relationships between the features and between some of the features and also the special feature or the special column to be predicted. And we don't want to say that the features have to cause the label to be predicted or the other way around. We have uncertainty over that. So what we did is sample and initialize a causal graph, sample edges in the graph, and then sample where in this graph might be our x1, where might be our x2, where might be the y, and the inputs, they are noise variables. And so this here, in this case here, is not used actually. That's perfectly possible. And we sample connections and also sort of activation functions in there. And so this defines a function. And we can sample data points from this function by sampling these noise variables and then pushing them deterministically through the graph to get these values. And then we say, well, this here, that's supposed to be the y value. So that's the y value. This is the y value. This is the y value. This is supposed to be x2. So that's x2. And this is x1. And so we've built a data set that has 3 data points, and it's just one data set. And if we want to do classification, then we also have a thresholding here for binary. We can also do k-area. And then we, of course, do this not just with one data set and 3 data points, but we do this for millions of data sets with maybe up to thousands of data points. And in the prior, we also have a simplicity principle. So I mentioned that there can be variables in this structural causal model that do not actually cover x1 or y or any of the x's or the y's. So there can be these latents, and that allows us to model very complex functions and very complex relationships, but those should be less likely than simple relationships that do not have any latents in there. And so the likelihood of having these simpler graphs is higher than the likelihood of having these complex graphs. If we really have a lot of data, and if the data strongly suggests that there's probably a latent that's causing both x and y, then we can sample that. And then we can also model it, but it should have, well, we have a prior for having simple explanations. It's sort of Occam's razor. All right, and then we generate data sets from this, and these look actually quite similar to real data sets. So here we have synthetic data sets, sort of always plotting one feature against one other feature for, I guess, a total of 4 features here. And And here we have real data sets, and well, to the naked eye, I couldn't say which one is which. And that's sort of the point. We generate data sets that sort of look like real data sets, and we do millions of these. And for these millions of data sets, we learn to do forward pass predictions where we feed in the training data, feed in the X test, and output the Y test. And if we can do that, then the odds are that we can also do this for the real data sets. And indeed, this is the case. So here's a qualitative result, so very smooth and well-calibrated predictions. So here we have this classic half-moon data set from scikit-learn, and here we have TAPIFN predictions. So it's quite simple, quite smooth. Here we have sort of typical predictions by tree-based models that are much more axis aligned. Like a decision tree is purely axis aligned, and this is sort of an ensemble of different axis aligned methods. And yeah, you have MLPs are very different types of function approximators, and TAPIFN just gives you very nice and smooth confidence bounds, and it's actually somewhat similar in its predictions to more nearest neighbor models if you look at that. All right. Another big thing is simplicity. So we had heard about AutoScaLearn before. There was a project, with probably 10 people working for a couple of years. It was very complex. There was Bayesian optimization wrapped around scikit-learn. Then we did some meta learning to initialize a Bayesian optimization based on previous data sets. We built ensembles and whatnot. And yeah, it's a very complicated process in the end. And this TAPIFN, it's a neural network. You do a forward pass. You could put this onto a sensor, and then you have a new algorithm that is being learned on your sensor by running a forward pass. It's quite amazing. And yeah, so if it might be hard to understand what exactly is happening in this transformer, but the execution and the use is just trivial, and it's just, yeah, it's a neural network. We can use all the neural network tricks, quantization and whatnot. It makes them super fast. There's a big industry about making neural networks fast these days because of inferences of large language models and so on. And we can use all that and just make tabular prediction much better and faster. All right. That was the qualitative result and simplicity. Now the quantitative result as of 2002, we had better performance in 1 second, just a forward pass, than any other machine learning method or AutoML method in 1 hour on average. So these are average results. There are data sets where TAPIFN is not the best, but on average, it was sort of, yeah, actually, if you have a GPU, it's 36,000 times faster than AutoScaLearn. That was the best on these 87 data sets. If you don't have a GPU, it's only 3,600 times faster. So this is like 1 second here, and this is 1 hour, and this is 0.1 seconds. And also it dominated all of the AutoGluon, CutBoost, etc. However, in 2022, it was limited in many different ways. So we limited the size to 1,000 data points, 100 features, and 10 classes, and didn't consider categorical features, missing values, uninformative features, outliers. And one big issue was also a high inference time. So when you train a new CutBoost or a random forest or something, it's very fast to do the prediction. So it might be slow to find the right hyperparameters and then actually train the model. But once you have the model, it's just a tree. It's just you trickle down your data a tree. It's very efficient. And here for TAPIFN, we just always have our training data and our test data point. We push them through the network together. And so actually training and prediction is sort of just one step. And if you have to predict millions of data points, that would be slow. But if you want to predict sort of for one patient at a time, then this is really not an issue. All right. Let's go to what has happened since TAPIFN 2. So since then, we have looked at scaling up. We have a more efficient attention mechanism to support more data points. We have a change in the architecture to support an arbitrary number of features, and we had some inference speedups. But more importantly, we have improved the prior in many different ways. So we have trees in there now in the structural causal models. We support more activation functions. we discretize the categoricals in the priors already. So we really make categorical features a first-class citizen, and then a lot of engineering in order to get rid of all the possible failure modes. And then we also demonstrated foundation model capabilities. I showed this picture before, so remember this here. This matrix here in this visualization is this matrix. So we have some rows, like each row is a data point, each column is a feature. So we have 2 features here, and then one special column that is the predictive, the response variable. And we want to predict this entry. So what we did change in TopFN is the architecture. So it's not just a standard transformer anymore. It's an axial attention transformer. So we have an attention that is just across the feature dimension. So you have attention between these and between these, and between these and between these. And then we have attention between the samples. So, so we would have an embedding now for every cell in this matrix, not for every data point. And then when you have an embedding for each of the cells, then you can do attention like this and attention like this. And you can learn sort of from other data points what this feature is with other data points, or you can also learn from what the other features are for your data point. And and then there's a standard MLP, and then there's 12 layers of that. And at the end of this, we just transform this into this predictive distribution by having, again, these 1,000 output bins, or 1,000 to 10,000. All right. Qualitatively, TopFN can now fit a very broad class of functions. So if you, for example, have a sine wave plus a trend, it can fit that very cleanly. X squared, the absolute function, which is something that, a linear function could, for example, just not fit this. None of these. The MLP can actually do this. Here it struggles a little bit, especially then when you get to these step functions. That's where trees shine. Trees are, when you get a sine wave like this, it's going to predict pretty much constant. And is not going to interpolate like an MLP or TopFN would do, or like a linear function would do. Also X squared, it just predicts these very constant bins. That's very standard with trees, which works great if you have step functions, then it's perfect. And step functions are a huge problem for MLPs and of course for linear models. And so TopFN can do quite well there. It's not perfect, but quite gets it that this is a step function. Then it can deal with noise. So it knows that if there's a whole of data here, that there's that there's still a linear trend, that there's noise, whereas cat boost would just be very much its typical step function. And more importantly, also, if you have heteroscedastic noise, so there's larger noise for larger values of this feature, then TopFN can also fit this beautifully. It can also work well with strong multimodal predictions. So here we have a true function that is actually from sort of your classical interference pattern in physics. If you have this double slit experiment, then if you have 2 slits and you have the light going through, and then due to this the dual duality between a wave characteristic and the particle characteristic, you get this interference pattern. So if you have here the the position on the wall, no, actually here's a, how does it work? So each sample, there's a position on the wall here, and this is the slit size. So if you have a really big slit, then you're getting more into just a single mode. But if you have smaller slits, then you get more of these interferences, like one particle here, 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 and nothing in between. And TopFN matches this very nicely, whereas cat boost, well, sort of matches it, but it's just not as good as this at this multimodal predictions. And so this is just to emphasize, we're not just having Gaussian predictions, and this is why we do think this regressionist classification is the right approach, so that we don't marry ourselves to a particular type of parametric output distribution for our predictions. But it's non-parametric, can just adapt to the data at hand. All right, then we also did a big quantitative analysis. So for this, we used the industry standard, the OpenML, AutoML benchmark. That's the best available curated dataset collection. There's a subset of 29 datasets that fulfills our criteria. We have scaled up now to 10,000 data points and to 500 features, and we just use anything. So we don't care if there's missing values. We don't care if there is categorical values, outliers, uninformative features, etc. The goal is really to work for any data set up to now this size here, so 10 times larger than last time. And then we do tenfold cross-validation, report averages. And we have a single trained TAPIFN that is used in all the experiments. And that was trained actually relatively cheaply in 5 days for on 8 GPUs that are 6 years old. So these are really, anyone can afford this. This should cost, it costs less than $1,000 to train this. So yeah, it's not like training your big foundation model, Llama 3 or whatnot. All right, and the results for classification is that, well, TAPIFN does a lot better than other methods. In particular, the best other competitor method in our results was CutBoost. CutBoost in particular is much better than XGBoost in terms of its defaults. XGBoost can sometimes have pretty bad defaults that are not even better than random forest. If you tune XGBoost properly, then it gets better and better. So given more time, it gets better with the hyperparameter optimization, but still, yeah, TAPIFN with its default non-tuned is already better than XGBoost. With this is 4 hours of runtime, and this is like 5 seconds. So it's more now than a second because the data sets are larger, so it's up to 10,000 data points, and that's why it's taking a bit longer to predict, but 5 hours versus 5 seconds versus 4 hours. And in the default, so this is this default against this default, TAPIFN is better in 28 of the data sets. There's still one where it's slightly worse, that's entirely expected. It's not guaranteed that it's best everywhere. And now after tuning, yeah, CutBoost is on par on some, and then slightly better on 3, but still TAPIFN is better on most. And now we also have regression. We didn't have regression before. And there it's sort of a similar picture, but a little bit less of a difference. So TAPIFN is still the best, but the 5-second version of TAPIFN is sort of on par with the 4-hour version of XGBoost and CutBoost. And yeah, CutBoost is also stronger on more data sets, but on more data sets and for classification, but TAPIFN is better on more data sets than CutBoost is better. So overall, not quite as strong as for classification, but still clearly outperforms the baselines. Then we also compared to AutoGluon, which has really emerged as the industry standard for tabular data. It is the current best AutoML method. I'm not involved in it. I did AutoScaLearn and AutoWeka and AutoPyTorch. And AutoGluon is just much better engineering. Nick Erickson, the main guy, is a gifted engineer. And he really just tuned this to not use any poor implementations of different algorithms and so on. So the base level is just very strong there. And on the meta level, he's by now like incorporated everything that's in AutoScaLearn 2, and additionally has this multi-level stack assembling. He had some analysis that actually all the mechanisms from AutoScaLearn 2 were actually more important than the multi-level stack assembling. So if and AutoGluon originally became known just because of this multi-level stack assembling, but it was particularly strong just because of the of the strong base level. And now it's strong base level and all the tricks from AutoScaler 2 and all the tricks from multi-level stack assembling. And so there's just all the tricks in there, and it's just very strong. It's a dependency nightmare because it has a lot of different base level methods that it uses, but still. And it has pre-optimized hyperparameters, so it just basically doesn't do much of a hyperparameter search anymore, and that's something we had in AutoScaler 2. And the post-hoc ensembles, so it makes predictions and then puts these predictions into an ensemble of its own with this multi-level step. And we also here now mimic this in TAPIFN to also do post-hoc ensembling of TAPIFNs with different hyperparameters. So that's the tuning you saw earlier. So if you tune, you would tune with different hyperparameters of XGBoost. And the result here is, well, the win ratio of against autogluon is sort of 75% to 25%, so highly statistically significant. For classification, even the 5-second version of TAPIFN without this PHE is on par or better than autogluon, and with this PHE, it's clearly better. And for regression, it's the standard version of TAPIFN is not better than autogluon, but when you tune it, it's a bit better. And together with this PHE, it's again clearly better win ratio of sort of 66 to 33%. All right, so that's a comparison to autogluon. We had various failure modes before, so we did some analyses of those. So uninformative features was an issue before. So here in our experiment, we have, let's say, X features, let's say 50 features, and then we put 450 additional features in that don't matter at all. And that was something that TAPIFN1 would have big problems too, because neural networks are notoriously bad at handling these uninformative features. You see the MLP performance here without uninformative features, and with uninformative features, it's just crashing. And TAPIFN1 used to do that, and TAPIFN2 just doesn't care so much. Actually, it's more robust to it than CatBoost and linear models. All right, and yeah, even if you put these 90% uninformative features in, it's still better than CatBoost that is not misled by these uninformative features. Missing values. So here we looked at the subset of data with or without missing values. And on both of them, TAPIFN was strong. Linear models were a bit better for the ones with missing values. CatBoost and MLP actually also didn't work so well for the missing values. And all we did for this was in the prior, we have the possibility of features missing, and that's all it took in order to fix this. Categorical features, again here, so the setup is subsets of data, data sets with and without categorical variables. CatBoost is particularly strong for categorical variables. MLPs are particularly bad. Linear models are also not so bad. And TAPIFN is actually also even a bit stronger on the categorical data sets. And there it was just very much the work on the prior that helped us fix this. Another analysis, so TAPIFN really works well with less samples. And in particular, so if you compare TAPIFN with access to half the samples, is still slightly better than CatBoost if you give it all the samples. So if it costs, I don't know, $10,000 to do analyses on a new patient, and you otherwise need 10,000 patients, and you can here get away with 5,000, with half of that, then this could really help us, develop diagnoses in a much more cost-efficient manner, etc. So you only would need 50% of the data to get to the same performance as previous methods with 100%. All right, you can try out this new version. We have a tiny URL here. I think I won't do this live demo quite now. If we have time, then maybe we do it later. I'm trying to copy paste this. Oh, come on, into the chat window. Where is the chat? Don't see the chat. There's the chat. And let's go back to the presentations and wrap that up first. But let's wrap it up first. I also want to briefly talk about foundation model capabilities. So TAPIFN allows fine-tuning because foundation models allow you a foundation for other work afterwards. So if you pre-trained on this massive data, now you can fine-tune to particular types of data. So if you have this orange data, there's not that much data. So the blue curve is what TAPIFN would predict now, which is not perfect, right? It doesn't get this seasonality here because it doesn't know that this would be a time series or anything. It's just getting the X, Y values. And if you then actually fine-tune a little bit on sign functions, then...
Excuse me, I can't see the slides, sorry.
Yeah. Oh, you can't? Interesting. Let me redo the sharing. I didn't stop the sharing, but since I guess I left the window, and that made Zoom stop sharing. Here we are. Good. So that's what I was talking about. There's these. Hang on. You should see it now, right? Do you see it?
Yes, perfect. Thank you.
Right, good. So there's this X value, Y value. If we, as humans, look at this, well, it's clear this is a sine wave. This is TAPIFN without fine-tuning, and if you fine-tune, it gets this that it's supposed to be sort of a sine function. And you could fine-tune more and it would get better, etc. But so fine-tuning is one of the foundation model capabilities that it has. You can also prompt tune it. So where you actually have this training data, you could actually change your training data. If you have like 10,000 data points, you could say, well, which type of like subset of 1,000 data points might give me the best performance for these 10,000 data points. So you can change the inputs to the model, just like you convince GPT differently by asking differently. You can also then use this for bias mitigation. So you could actually have this multi-objective prompt tuning where you optimize for different objective functions, including algorithmic fairness. You can use this for interpretability. We did one study where we had like 1,000 data points and did prompt tuning and data set distillation are very similar here. So we actually distilled 2 data points that were synthetic, but if you put these 2 data points into the pre-trained TAPIFN, you could get just the same performance as you would get with 1,000 data points. And so it's a really nice interpretability measure to be able to say, well, these are the important data points, or like it's very much like an SVM, or like these are the important this is where the important information is, and with these 2 data points you don't actually need much more, and the inference time then also goes down if you have 2 data points only that go forward through the model together with your new test point, then inference time also goes down dramatically. The details on the bias mitigation. So here's standard TAPIFN. If you optimize with prompt tuning for accuracy, you're going to improve that. But if you optimize for both accuracy and there's a demographic parity, then the demographic parity improves everywhere. All right, you can also do counterfactual reasoning with TAPIFNs. This is maybe a bit too complex right now, but counterfactual reasoning is sort of these analyses. What would the result be if I changed a protected feature, and a protected feature like gender, like what would happen if this patient was a male, not female? Then you want to remove some causal effects of the gender on the other features. Maybe in medicine this doesn't apply so much, but in college, like in college applications or credit scoring, etc., you actually are by law, the law demands that you don't discriminate against gender or bias, and then just taking out the one feature, gender, that doesn't help because there's a lot of correlates with this feature, like the size of the person, the weight of the person, etc. And you really want to remove the causal effects that the protected feature has on other features, and you can actually do this with a TAPIFN. Maybe if someone cares, I can talk more about that later or offline. But it's quite neat. You just basically change the prior. And then again, just do a forward pass and tell TAPIFN which is protected attributes. And you can also do drift resilience with this by actually having in the prior data another sort of time component that you change over time, and then you can also say, okay, it gets much better if it knows that there might be concept drift. All right, there's a lot of questions that remain. In particular, substantially faster inference times would be nice. Andreas Müller has already done great work on looking at hypernetworks, where basically your TAPIFN-like model outputs another MLP, and then you use that MLP in order to do predictions on a test, and you backprop through that MLP and back to your TAPIFN in order to improve the predictions of which MLP to use in order to really have the best MLP for new data sets. So you basically look at a data set and say, for this data set in a forward path, I'm going to use this MLP, and then just predict with this MLP very quickly. MLPs are actually faster in prediction than the XGBoost, etc. So there's a cool work on this. Unfortunately, this doesn't work so well yet. And I think it's important to follow up there. Then crucially, understanding is lacking. Like, how does this learned TAPIFN algorithm works? This might also teach us something about, in general, how does in-context learning work? How does GPT work? And there's all kinds of mechanistic interpretability studies you can do. In particular here, you can do a lot. You can dissect how this algorithm works because training is fast. Like, our typical trainings take like a day on one machine. For this one production run, we had 5 days, but and then so we can scale up and down model size, see how that affects everything. We can use just a linear function and see how does it map this into the transformer. It will learn based on linear regression, but how does it do this? You could have a prior of step functions. The network will adapt to this very differently. And so you can really look at, okay, with this prior, what will it do? How does it do this? And then so there's a lot of really cool research left to be done. Also study this whole space of pre-training, fine-tuning combinations. And then finally, it would also be very interesting to approximate true posterior inference over the latent model parameters. So I mentioned many times that we bypass that step of modeling the latent, but actually it would be really nice to have a posterior over what is the right structural causal model for explaining this data. What's the probability that this feature here causes this? What's the probability that this feature here causes my disease? And you could really have sort of priors over, well, probably it's like this, but here I'm uncertain. And then you can get posteriors over these priors and actually really learn more about how diseases happen and so on. All right, so let's get to the takeaways. TAPIFN's remaining limitations are being removed. The goal that we currently have is sort of a new default algorithm for tabular data for up to 10,000 data points and 500 features. We do hope that we have achieved this goal, and you can already try it out. Please try it out and let me know if it works or if it doesn't work. TAPIFN is a data-centric approach and really user-centric. Can exploit your knowledge about your data. We can make a workflow in creating better TAPIFNs, or where you just say, well, these are the types of data sets. We push a button in order to create a new TAPIFN for that. And you can get really quick iterations with the user because it's so fast. You could sort of think of a web interface. While the data uploads, you already get some predictions. And so we really want to move more towards interactive data science with this as well. So TAPIFN is really on its way to become a new default for supervised machine learning. And it's also just one example of PFNs. I also encourage you to try PFNs independent of tabular data. With that, I'd like to thank you, and I'm available for questions.
Thanks so much for that great talk, Frank. That was very interesting and illuminating. So for questions, I think how we're going to do this, we're just going to, I think some questions have been posted on the chat. I think we'll just go through them chronologically to make sure we don't miss anyone. So maybe what I will do is I will just call on people who have asked questions so they can unmute and ask you directly. Yeah, so maybe we'll start with Sarah Aliko. And sorry if I'm mispronouncing anyone's name. I definitely will be doing that.
Hi, thanks for a very interesting talk. I'm not very familiar with Bayesian networks, so this was really nice to hear something new. So my question might be slightly uninformed, but it's regarding the training of tab PFNs. My assumption is like you would want to create a prior space where you have as many functions as you possibly could cover so that you can have this one-size-fits-all model, right? So if that is the case, and obviously there is limited compute and time in the world to do everything, having too many training data points for a single of those prior functions, does it not cause overfitting by constraining this number of functions, or how do you deal with that?
Well, so there's two things. Overfitting is usually you overfit by optimizing hyperparameters, etc., on the actual data set that you see. So like you get a new data set, there's 1,000 data points, then you need to think, okay, how do I divide this into maybe a training data set and a test data set, or a validation data set, or you do some outer cross-validation or inner cross-validation or nested cross-validation. But it's all about what do I do now with these 1,000 data points that I have. Whereas we just never do anything on these 1,000 data points. We do stuff before, and then when we get these 1,000 data points, we put them into the model, do forward pass, predict. And so that avoids overfitting, looking like doing something special for the data set at hand. And how do we avoid overfitting to a particular type of function class? Well, I mean, we can sample a lot of different functions, and we sample like 10 million or something like that. After that, it seems to quite converge. It is possible that our prior that we've built with the structured causal model and so on, that this misses some types of functions, and then we will not predict as well for that. That is a possibility. We haven't seen that so much in our evaluation, but yeah, you can go and do, and this is actually something I really want to do. You could go and generate adversarial data sets for which TAPFN is arbitrarily poor, and then maybe also go and put that into the prior and say, well, you should also be good on that, and then it will just start to get better on everything. There is a thing, there is like no free lunch theorems, but actually for realistic functions, for useful functions, they don't really apply. Of course, once you have data sets that say, well, my training data is like this, and then the test point is here, and actually this is the right prediction. I don't think we very much care about this. If this is sort of an adversarial example, then okay, I'm happy with this being an adversarial example. I wouldn't expect that to make that prediction. But yeah.
Thank you.
Okay.
Great. And next, we have a question from Samuel about calibration.
Hi, thanks a lot for your talk. I was just wondering, do you look at the calibration performance at all in terms of the models, or are you just focused on the discrimination?
We've also looked at calibration, like sort of expected calibration error, etc., and it was also better. It's in the appendix. So haven't showed these results, but yeah, it was also better across the board.
Cool.
Right. And Seraphima, if I'm pronouncing that correctly.
Yes. Hi, thank you for a great talk. I'm not, I think my questions changed during the lecture. So I'm not sure, I'm a bit confused. Like on the one hand, you say that the methods are already pre-trained in all kinds of distributions and functions. And on the other hand, you do need a prior for my data. So let's say I have a small data set, like 10 points, like you mentioned in the beginning. Is it enough to really like use
So I guess I wasn't clear enough. So we built this once on a particular prior. If you say this prior is just a dumb prior for my type of data, I know my data, I know much better what my data is like, then you could build a better TAPIFN for your data. But in order to run this TAPIFN, you don't need to tell it a prior. You just need to tell it your 10 data points and it should do its thing. But you could make a better TAPIFN by actually telling it your prior.
Okay, so if I do have an understanding of the distribution of my data set, I can again run the pre-training set and generate synthetic data sets, right? And then it will be better.
Yeah, so you could, for example, and this is something that could be of interest to people in the audience. I mean, because the medical folks with knowledge that is super valuable. And if you have this sort of valuable information about what do I think are actually the possible structural causal models for my data, then you might have sort of this mechanistic model that has some question marks in there. Here, it could be that this causes this, that this causes this, or it could be that this causes that this causes this. So then, then you just have 2 or 5 or 10 structural causal models. But so you could build a prior for your particular case where I'm like, well, I know that this causes this. I don't quite know how strongly it causes it. So let's put a little prior on there. Then I know that they jointly cause this. I don't know how that relates to this other thing. So I need to put uncertainty there. We don't have a great interface for you to do this yet. But, but this is something that I think would be really what we do want to do in the future.
Thank you so much.
Thank you.
Right. And I think George, if you're still around, I think you had a couple questions.
Thank you very much, Frank, for the great talk. One is related to kind of, that's a little bit selfish because I'm working on time series data, but are you planning or are you working on kind of time series based variant of your kind of approach based on PFNs? In order to model kind of the temporal dependencies also in the data.
Yes, we do. We shall submit a workshop paper next week. Yeah, so in a nutshell, yes. There's many different types of time series. So there's this univariate time series. They are very straightforward in a sense. And then there's multivariate time series that are, and for univariate time series, we've seen a lot of progress. There's a Kronos model and other models like that. But multivariate time series, I was talking to Alexander Schur yesterday, who is one of the authors of Kronos, and he actually said, yeah, if you have multivariate, my best bet is TAPIFN, and in some sort of TAPIFN with temporal dependencies, etc. And that is also what we're seeing.
Perfect, thank you. I have a second one, which is related to explainability of your approach, because your approach kind of learns the model within the weights of your model, kind of the transformer, right? So on which level of, can you provide explanations for the things that you train, kind of?
Mm-hmm. Yeah, that's a very good question. So, I mean, we can clearly use all kinds of post hoc interpretability tools that you could use for any black box models. We also have another approach where that's very much like this hypernetwork stuff from Andreas Müller. It's actually collaboration with Andreas Müller, where we output a GAM, a generalized additive model, And so then it's really just this very simple additive model where you just take this feature times so you have these functions. This is my relationship for this feature, and then you add the relationship to this other feature. So it's just extremely interpretable. It's also with Rich Karana, who is one of the typical generative additive model persons. So you can use this mechanism to fit GAMs better than fitting them from scratch and avoiding overfitting and so on. And similarly, you could probably also output multi-adaptive regression splines or whatnot. But on another level, you can also use this to do the typical analyses that you do with any type of black box model. Like, for example, SHAP in particular, we have looked at, and SHAP analyses of our model seem to be much more consistent than SHAP analyses of weaker models, because then there would be just a lot more noise in those. And if the model itself is actually stronger, then the SHAP explanations were actually also less noisy. And so, yeah, on this sort of individual level, it's sort of quite easy to give you these types of analyses. What is more elusive so far is, how does it learn what it's learning? At which level of the architecture is which feature formed, and so on. That there, a decision tree is easy to interpret, but because of that, people then typically think, oh yeah, and then XGBoost is easy to interpret. But XGBoost fits like thousands of different decision trees of depth 100, and there's just no way to interpret this. And if you look at the predictive distribution of XGBoost, it's just super jittery and not smooth at all. And so yeah, an individual decision tree might be interpretable, but the overall thing might be very uninterpretable too. I think there's many questions left to be answered.
Thank you very much.
Thank you.
All right. And I think Miriam, you had a question, if Miriam is still around.
Yes, hello. Thank you very much for the talk. It was very interesting. I was wondering, in the case we have tapped PFN for example, like a medical data set with tabular data that is clinical and demographic, and then you contain like different types of variables, categorical, binary, and regression, and how does it deal with these types of different variables? Because it's very difficult to extract the same like information when you have like binary and regression. Thank you.
Sorry, I don't think I fully understand. How does it deal with it? I mean, you just categorical variables, you just feed in directly as categorical?
Yeah, but then like it treats all of them the same. Like it doesn't make a distinction, I understand. Like it's the same, like you don't distinguish between categorical variable and then regression variable. You just feed them like you would feed like to a random forest or a XGBoost.
Yeah, I mean, so you have an embedding that's different for your categoricals and for your continuous variables, but like an XGBoost, you just tell it here's an input is categorical, and for TAPFN you tell it here's an input is categorical, or here's an input it's
okay,
numerical. And
okay, thank you.
And it deals with it natively now. And before the version 1, it was very poor. It would just like have a category 1, 2, 3, 4, 5 and just feed it into the network as the number 1, 2, 3, 4, 5. And it's not doing that anymore. So it's not as stupid anymore. Okay.
Great. I think that's all the questions we had in the chat. I think we'll open it up just to everyone on the Zoom. If you have any other questions, maybe just like raise your hand or just on.
I could also, if you're interested, do a quick walk through this demo that I mentioned.
Oh yeah. Yeah, it seems like we don't have any additional questions at this point, so the walkthrough would be a good idea. Thanks.
Yeah, so I put this tiny URL. Did you see my screen?
Yeah.
Right. Yeah, so this tiny URL I put into the chat earlier. I'll just put it again since I'm here. so you can go through this yourself and try it. Yeah, so like first it's just the pip install. So it is on PyPI. It should be quite straightforward. I think it will prompt you to restart your session because that's a call-up thing. It always does that. Then we install some baselines. There's some imports for the plots and so on. And then, so this is something we don't have this available yet, and it's faster on GPU. But what we do have is we actually have a server running in the cloud, and we give you GPUs. So you can actually run it there without having a GPU yourself, but you need to make an account for it. So this init function now runs through for me, but for you the first time it will say, well, are you an existing user or do you want to make an account? And then sends you an email and then you get your account. Of course it's free, but yeah, we need to limit sort of the number of users on these GPUs that we pay for from university money. And anyways, also please tell me if there's anything wrong with this, because it's the first time we're building something like this. So yeah, if initially the emails went to spam, by now they shouldn't go into spam anymore, but if it doesn't work, maybe check your spam folder and let us know that it still goes to spam. Anyway, so let's continue with this demo. So this classification data, we just load some data. This is from scikit-learn, breast cancer data, a very standard data set. We predict with the baselines. This takes sort of 20 seconds or so. This is just with their defaults. And yeah, then yeah, you predict with TAPFN faster and compute the performances, and surprise, TAPFN is a bit better. And this might seem like it's very similar, and that's often the case. You know, it's different machine learning methods tend to be quite similar. Here in this case, XGBoost, as I mentioned before, with its default, it can be quite poor, but yeah, it's not too uncommon to get like 99.6% with this and 99.5% with that. That's sort of that's still considered substantial, depending on the application. All right, then we have regression. We have the diabetes data set. So, and this is all meant to be like, you can just put your own X train, Y train, and so on, and see if it works for that as well. So diabetes data, you can have this TAPFN regressor or XGB regressor, CutBoost regressor, random forest regressor, and then compute accuracies, and then yeah, there's some toy functions that are maybe more interesting because they're sort of visually interesting down here. So you have a random forest classifier and XGB and TAPFN classifier in these plots, and so this is something that is sort of, yeah, not very easy if you don't have the right features. If you sort of had a feature, how far am I from this point? Then this would be quite easy to predict, but we don't have that as a feature. We only have the XY coordinate, and so the forests make these very axis aligned choices, and TAPFN sort of makes it visually more useful predictions. And then we have a sign function here. Yeah, get these predictions. As I showed before, sort of random forest will not do this interpolation. And then you can also do this quantile regression. The data set that I have here is not this double slit experiment, but it's sort of this heteroscedastic noise experiment. Yeah, so you see this heteroscedastic noise in between the data points, and then we have an example where you just downloaded a picture from Batman, and so these are the input data points, and this is what XGBoost interpolates, CutBoost, and TAPFN. But that's more a little joke. But yeah, anyway, so that is the Colab, and hopefully you could also just put in your individual data sets and see if it works for that. And if it doesn't work for your data, please let us know. He says, oh, and you can also, during the sign up, you can tell us what you do, what sort of your background is, and if we can email you when we have new versions, because we are developing this heavily right now. Right. Yeah. Any questions? What I expected to work well with large tabular data. Well, so this web interface, if you have more than 10,000 data points and 500 features, it will just say, I'm not going to do that because that would use too much resources right now. We have various works for looking at how to scale up. Because I mean, if you have a really strong method that works for the core of what machine learning on tabular data is, then you can wrap all kinds of things around that. You could think of a k-nearest neighbor that says, well, let's take the nearest 1,000 data points and fit a TopUFN on that. And then people have done that. Yeah, we actually looked at this 2 years ago, and we have something better right now, but that's not out yet. But yeah, you could think of many different ways of using it inside of a bigger thing. You could also, of course, ensemble different models. I have a TopUFN that's fit on these 10,000 data points, one on these 10,000 data points, and I ensemble them in different ways, maybe depending on the uncertainty for the test data point. So there's many different ways like that. Right now, there is no existing version where I'm like, yeah, 4 million data points, this is going to be awesome. We're working on it. How does TopUFN work with survival analysis? So we like if you basically feed in, well, these are the features, this is the predicted time. There's no, like, you could think of making a special prior and so on with some Cox model, and actually the first author of TopUFN is a med student at Charité, and he is or has developed a survival model along these lines. You could use the existing TopUFN and just feed in, well, these are the features, this is expected time, but it might also predict something. If it is not constrained, it might predict something that, I don't know, the predicted lifetime is negative or something like that. If it's not constrained, it might do that. But if the prior constrains it, it would not. The email I should also give you. There's the email. Good. Are there many more questions? Did I miss any questions?
No, I think you got all of them. Yeah, you also timed that perfectly because now we have to hand over to the next session.
Okay.
So yeah, thanks again for the great talk. And I guess I assume people will be reaching out to you about the really cool work.
Mm-hmm.
Cool. So I think,
and then if you're excited about this sort of stuff, we're hiring. We would like to also see collaborations with practitioners, also in the industry or something. We just lack the right amount, the right data. So if you have cool and exciting data sets, please do get in touch as well.
Great.
Thank you.
Okay. I'll hand over to Max and Ari now for the next session. Thanks everyone for joining.
Thank you.
Hey. Hello, everyone. I believe Kasia should also be here.
Yeah, I'm here. Okay, so the next speaker in our series is Dr. Ercole, who is an affiliated associate professor at the Department of Medicine in University of Cambridge. In addition, Dr. Ercole is a consultant in neurosciences and trauma intensive care at Cambridge University Hospital's NHS Foundation Trust. Dr. Ercole has an extensive background in physical and statistical modeling, as well as data science and computing. And his research involves development of novel medical technology and its application to the treatment of clinically ill patients. In today's talk, the talk is titled From Bench to Top Real World, Challenges in in implementing digital healthcare. As we all know, healthcare is a highly regulated environment. And while there has been a huge growth in interest in digital healthcare, very few AI algorithms make it into clinical practice. In this talk, Dr. Ercole will explore the challenges involved in real-world implementation and evaluation of AI systems and algorithms in medicine. He will describe the regulatory framework for the reuse of real-world data for algorithm evaluation, which is essential to avoid legal difficulties, and also the behavior change challenges that come with real-world deployment. Professor Ercole, over to you.
Thank you very much for the introduction, and thank you also to the organisers for the invitation to speak. I'm quite looking forward to this because this will be a rather different presentation to the sort of presentation I normally give. Just a little bit more about me and explain where I'm coming from. As you quite rightly said, I'm an intensive care consultant. I've been doing this for quite a long time. But I've also done a lot of data processing and analytics, and I've had an interest in machine learning and informatics for a long time, particularly in intensive care, because that's my main domain. But there's a few other bits and pieces that I will bring up over the next little while. I'm the principal investigator for something called ERIN, which I will talk about later as an overarching ethical framework, and allow me to talk a little bit about how we get hold of routinely collected data. I'm the Chief Clinical Information Officer for Cambridge University Hospitals. That means I provide digital leadership in terms of actually how do we implement and use digital technologies in clinical scenarios. And then finally, I'm one of the clinical safety officers for the hospital. So I'm involved in the risk assessment and assurance of digital tools in clinical service. So this is going to be much less about computer science, but much more about software engineering, about regulation, and about real world. And I need to start with a caveat, which is that regulations obviously vary significantly around the world, and I cannot cover all jurisdictions. So I'm necessarily going to take a very UK and, to be honest, EU standpoint. The regulations in the UK are very similar to those in the rest of the EU. But similar sorts of concepts apply around the world, for example, in the US and so forth. Right, so chapter 1, I want to just think a little bit about where we're going, where are we coming from, and what the medical AI development life cycle really looks like. And I'm going to begin with a deliberately extremely provocative statement. And the coolest, smartest, best performing, most revolutionary, most novel medical AI model is utterly useless if it doesn't change clinical practice. I'll just let that sink in a little bit. I'm likely to be drummed out of the boys' club now. But it is true, and it's not just AI. So this is the molecule. I don't know if anyone knows what this molecule is. You can extract it from the willow tree. If only people used it after heart attacks around the world, we would save millions and millions of lives. It's aspirin. We know it works. It is a very, very cool molecule, but people still don't use it, and therefore people still die unnecessarily after they have a heart attack. So you have to bring things into clinical practice, and people actually have to use it. Okay. I want to just, before we think about AI specifically, I want to just now take a slightly different view and look at how we develop medical interventions in general. And mostly this is really the model for drug discovery. And then I'm going to overlay and think about how machine learning and AI might fit into this model. So the model typically starts on the left-hand side with basic research. That's understanding physiology, it's understanding biology and so on. Then some early discovery, some preclinical work not in humans. Then we move into a phase which is the clinical development, and that's normally split into three components. Phase 1, which is really a study which looks at, is this safe? Is it toxic? Phase 2, which is, does it actually work? Does it have the effect that we think it's going to have? And then phase 3, if it has that effect, is it actually better than anything else that we've got? Is it worth doing? And then we move on to a sort of post-marketing monitoring period once the device has been deployed into clinical practice. Now, if we look at this diagram, first of all, the clinical development part, this involves real patients. That's the thing that makes this different. Okay, so that is, phase 1 studies typically involve tens of patients, take months to do. Phase 2 studies typically involve hundreds of patients and may take years to do. And then phase 3 studies, the final, actually, is this something that's going to change clinical practice, are typically involve thousands of patients. It may involve many, many sites. It may take many years to do. And as a result, if we go up to the top, the basic research is in fact relatively cheap. But as we get into, and there are many places doing basic research, but as we get into clinical development, the number of drugs that act, or molecules that end up entering that system is smaller. So there are fewer phase 1 studies than there are early discovery and basic researchers. It is much more expensive. And as we get through to phase 3, it becomes more and more and more expensive. Okay, and there are fewer and fewer. We expect an attrition of molecules as we move through this process. The other thing to sort of overlay on this is that in the early stages, the risk of patient harm is 0. And that increases as we go through clinical development and actually continues when a drug has been released into the wild. But the risk of failure really, is really significantly higher at the beginning. In other words, I would expect many, many, many early therapeutic, early discoveries to fail and never make it through preclinical or clinical development. Okay, so that's the situation for drug discovery. And then the final thing just to overlay on this really is that academia probably plays a large role on the left-hand side of this. And then ultimately, we always have to move into sort of industry because we are looking at large amounts of money and for deployment in industry. Okay. Now, very similar things, very similar concepts apply to the life cycle of a clinical or a medical machine learning algorithm. Okay, so we have basic research into machine learning and computer science. We move through to early discovery and preclinical, and these are typically the sorts of bits of work that are done with maybe open access databases, things, code that's cobbled together in Python, in notebooks and so forth. And then, but in order for that to make progress into the right-hand side and ultimately into clinical practice, we have to ultimately be aiming towards a clinical development pipeline. And that means that we have to actually deploy a piece of software into a clinical environment. And we have to get clinicians using it as part of a pipeline, a pathway actually within that system. And this is not easy. This is a difficult thing to do. And yet, and I suppose really what I'm going to be talking about here, I'm going to be talking about this part of it, the focus of this talk, given the audience. But really the point I want you to come away from is that you need to have, it is very helpful to have the next bits of the pathway in mind when dealing with the early parts. Okay, because otherwise there is a risk of answering a question that no one is asking, or getting to the end and finding that there's an enormous gotcha that hadn't been considered early on in this pipeline. So that's really where I'm coming from for this purposes of this talk. We know there is a significant translational barrier for machine learning models. Now, this is a piece of work we did in the intensive care unit. We mapped machine learning publications, predicted models, it's a few years old now, to the NASA technology readiness levels. And as you can see, 93% of them are stuck in TRL 3 and 4. So analytical or experimental critical function, proof of concept, component, breadboard validation, that kind of thing. Very, very few systems actually make it through to mission deployment. Okay, now that's expected, but it is a problem, and it would be good if we as a community could take steps to try and address this. So the last thing to say about healthcare research, which you are all in if you're interested in machine learning in clinical practice, is that medicine is a really highly regulated industry, and that is just a fact. So I am regulated as a doctor. I can't do whatever I like. Drugs and therapeutics are regulated, and therefore software, and one of the things I'll come back to later is software as a medical device, is regulated too. And it's right that it should be regulated, because you do need to consider if your AI model, when it's deployed, misbehaves, then genuinely people may die or may be harmed. Okay, you may miss a diagnosis, you may fail to triage a patient appropriately and therefore delay their treatment, which could kill them, or you may make incorrect treatment decisions. And then perhaps slightly less critically, but important at a societal level, we know there are biases with training data sets. So there's a risk of actually worsening healthcare inequalities, which is a concern to the healthcare community and to society. If your software misbehaves, and I'm not going to talk a huge amount of this today, then A, it may not be available when it's needed for treatment decisions because it's broken today, or B, it may cause a healthcare risk or a cyber threat or some other deployment risk to the healthcare to the community. And then finally, and this is very important to understand, if your doctor or clinician misunderstands what your model is meant to be doing, then they may very well use your AI model for a purpose it was not intended and therefore is actually unsuitable for. So nothing wrong with your model, but it's being used in the wrong way. And these are all considerations that are worth bearing in mind right from the very outset when defining your problem. So first thing to say really in terms of what to do is there are now plenty of mainstream guidelines and research methods and reporting for early sort of preclinical work in AI. And you should all, if you're not already familiar with these, become familiar with them. They are important, and it's very difficult to get anything published in the medical literature without applying these statements. Okay, and again, I come back to my deliberately provocative statement, but the purpose of this talk is to aim you stop, what I'm aiming to do is to stop you wasting your time later on. Okay, so really, really important to consider the development life cycle at an early stage. And again, I just need to say that there is a balance here, right? I expect many early machine learning, clinical machine learning projects to fail. So there is a balance between doing a lot of excessive work for projects that are likely to fail versus not doing enough, not taking enough care, and therefore progressing a long way down the road before finding some sort of showstopper. And crucially, and this is really what this talk is going to help you, I hope, do, it's important to move to test on the best possible and real data that you possibly can, not just the data that you have easily available. And I'm going to walk you through in this talk the regulations surrounding how to do that. I think it's really important. The second thing to think about is where this fits in, where your model fits into an actionable workflow. So, okay, you may predict something or diagnose something or cluster something, but where will this actually be used in clinical practice, at least approximately? Because that definition of the functional requirements at an early stage is crucial to define the acceptable performance of the model that you're going to build. And ultimately, the third thing to come away from this talk realizing is that you will, if successful, your software will lead to, will actually end up being a medical device. It will be software as a medical device. And some kind of at least awareness of the regulatory framework is important, I think, from even at the outset. Otherwise, it becomes impossible to unpick these things without completely rewriting the code from scratch, which is expensive, time-consuming, and painful. So, chapter 2, let's talk about how to get data from electronic health records. So real data, best possible data, the data that you really want to use to train your model to make it likely to really work. So you will be familiar, there are lots of data sets that are out there that are freely available, publicly available, either freely or with minimal regulation. Mimic is a data set that has been used again and again and again because it is very rich. From the ICU. There are other ICU data sets, including the European one, which I was part of the consortium. Plenty of these data sets out there, but they are limited and they are very specific. What we really want to do is get real data to address the real question that we're interested in. And of course, then we run into a number of regulatory issues that actually are not nearly as bad as people think. GDPR is held up as some kind of evil monster that prevents people from access to data for research. That is not true. In fact, GDPR is extremely permissive and in fact very, very helpful for researchers that want to use data, provided that we all obey and abide by certain rules. If we don't do that, then there are some real risks. People will know about the privacy concerns from Facebook, looking at people's social lives, but the Royal Free Hospital and NHS Hospital was working with Google DeepMind, doing a completely reasonable project that was ethically approved, but there was mission creep and mission scope creep, and the amount of data that they were requesting for wasn't well defined and increased, and ultimately they broke the law. There are implications as well for the greater good of research and the use of data within the NHS. You may remember this NHS data grab. It wasn't a data grab at all. It was really just an attempt at making sure that we all had access to data, but it was terribly handled from a public relations point of view. And as a result of that, this is the national NHS data opt-out data across all of England. And you can see at the point of making the headlines, the number of people who opted out from their data being used in research increased absolutely dramatically by about 10% or so. That is catastrophic. We cannot afford for this to happen again. So we have an absolute need to treat data carefully so that we don't create scandals and undermine public confidence. Provided we don't do that, the public is, generally speaking, very happy for us to do research with their data. So broadly speaking, the principles of medical research, and I should say there are exceptions to these, but number one is you cannot experiment on people without consent. More relevant to us is that you cannot use personal data without consent. And we have to understand that consent is very unlikely to be feasible for a big data project. If we need tens of thousands of it, we are not going to get consent. And therefore, we need to concern ourselves as a data science community, a medical data science community, to in this space to de-identify data. Okay, and once data is de-identified, it's no longer personal because it does not relate to the person. Therefore, does not need consent, and the GDPR does in fact not apply at all. So that makes things really simple, but I do need to talk about what de-identified means in some length, because de-identified itself is not a straightforward concept. Okay, and there is still some regulation that does need to be respected. So a fraction of this talk is now going to be talking about what de-identified really means. But broadly, we must handle our data sensitively. If you're involved in this, you need to handle personal data particularly sensitively, ideally communicate the benefits to patient public and not take data for granted. Okay, so what does de-identification actually mean? We talked about GDPR. If we de-identify things, it's no longer the only issue. But it's not the only constraint. Other EU member states may impose additional restrictions on GDPR. In the UK, we have a common law of privacy. So there are other things that need to be thought about. Okay. In the UK, these are the bits of law that may be relevant. There's, in terms of us regulating confidential patient information, Data Protection Act, and that incorporates GDPR. But as I said, if we de-identify, no longer an issue. The National Health Service Act 2006 gives a legal duty of medical confidence, so we cannot release things without patient's consent. But again, there is a mechanism for doing that, even if it's not identifiable. If the data is not identifiable, then it doesn't matter anyway. The way that research happens in the UK is that you initially go and discuss an idea with a member of an hospital R&D team. So you need local sponsorship. That's typically an NHS hospital. Then typically what you would do is go and get ethical approval. And in the UK, that's a national service, NHS Research Ethics Committee. It's a very, very long form, but not particularly difficult. There's additional research approval from a group called the Health Research Authority, and that's normally as part of the same ethics application. And if data needs to be obtained that is temporarily identifiable, for example, for linkage, then there's something called a confidential advisory group that will help as well. That sounds really hard, but for two things. First of all, it's not actually that difficult. This was it's not a very exciting intervention study, but Amelia Robinson was a medical student who came up to me and said she wanted to do a quick project and could she do a randomized a trial that was going to require ethical approval. And I said, well, are you sure about that? Because it will be quite a lot of work. And she said, yes. And we pulled this off when this required full ethical approval, we pulled it off in a matter of months. Or in fact, Amelia did. So this is not difficult. Even better than that, the other bit of good news is when we're working with de-identified data, technically in the UK, we don't need ethical approval, a rec submission. So that reduces the amount of paperwork hugely. It does need HRA approval, but that's actually generally very straightforward. And the HRA normally almost waives data-driven stuff straight through. And it does need some kind of sponsor. So it will need to work with a hospital or the data controller or whoever the data is coming from. And what the data controller will want to be sure about is they want some assurance that the data, when you've got it, is secure. And that will include not just the physical location of the data, but where the location of the cloud data storage is going to be and where the compute is going to be. They all want to know that it's not used for any other project and not going to be shared with anybody else for reasons I'll come back to later when we talk about the identification. And also that it's not shared with other machines. So, for example, that we're not putting data into a large language model that is somehow sucking up and hoovering this data and using it for something else. So those are the things that they all want to see. But this is not a difficult process. In Cambridge, for example, we've made it even easier. We've put a wrapper around this to speed it up. It's called ERIN, and I'm the principal investigator. It's the HR Research and Innovation Database. An ethical wrapper. So we've done the ethics for anyone that wants to do research. All that the investigators need to do is apply to a data access committee, which is local with a project description. And the data access committee, we will meet monthly. We assess the feasibility. We assess the risk using a tool that I'll show you in a minute, including the anonymization strategy. And if successful, that covers off your HRA approval as well. That's all that needs to be done. At that point, the researcher works with the database engineers to actually create the data extract, and typically are expected to work in the University of Cambridge secure research compute platform, but that's not necessarily necessary depending on the risk. Okay. But it is really, really straightforward to get hold of real data, and I commend people to try and do that. In terms of how we assess the risk, we recently published our data governance tool, and you can see the link here. But we assess both legal considerations, governance considerations, and reputational considerations to the organisation when looking at people's applications. But it's not a difficult process. Have a look through that, and you'll see the sorts of concepts and principles that we look at. So now, hopefully, I convince you that actually it's not difficult actually to work with real data, provided it's de-identified. But of course, that then begs the question: what does de-identified actually mean? Well, I'll tell you what de-identified does not mean. Anonymous does not mean, and this is commonly what people think, but it doesn't mean that it's impossible for a record to be mapped back to a patient. Okay, that would mean that no patient record could ever be unique. And that's not just hard to guarantee, but it's kind of statistically impossible to achieve in reality, right? Let's just think about 7 days of daily hemoglobin values, right? Which are integer values over a range, maybe 120 to 180. Assuming, and this isn't true, but they're uniformly distributed, like we've got 10 to the 12 unique combinations immediately, and there's only 10 to the 8 unique names in the world, right? And real medical records are much more complicated than this. So this is not what anonymous means, but it doesn't matter. Right, the fact that it's possible to match back to an individual does not matter because you don't have the original record, so it's irrelevant. But yet, this is what most people think that de-identification means. What it might mean, most countries that define this have a legal definition which says something along the lines of de-identified means a low probability of re-identification. Now, that's very nice, but unfortunately doesn't make a lot of sense because it leads to two questions. First of all, what do I mean by low? And secondly, what do I mean by re-identification? Okay, so this is all true, but it's not actually helpful. So let's think a little bit more about a mathematical framework or a conceptual framework as to what the identified really is. So here is the definition. I need to define a few different terms first. First term is an identifier. That would be like your name or your national ID number. Now, clearly, we need to remove all of that. So that's relatively easy. The second important thing for me to define is something called a quasi-identifier. So that is something or some things that could be traced to an individual if you had other data. So, for example, a postal code in the UK may easily identify a single household or an individual. So if you knew who was living in various houses, then you could put the postcode together and you'd know who the medical data was, right? So that's a quasi-identifier. And then hopefully there's a small set of those. And then the thing that we're actually trying to protect is the sensitive attribute. It's the thing that we're actually not wanting to disclose. So, for example, someone's HIV status. And so now the definition of de-identified is that we ensure the probability of linking a sensitive field to an individual through their quasi-identifier is low enough to be publicly acceptable. So that's really how we operationalize our de-identification. Now, some very important things flow from this. First of all, it requires a certain amount of public consultation. But in fact, the public are generally, when have things explained to them, are generally happy. But it also depends, and this is very important to understand, the probability of re-identification depends on the information that the researcher has available to them. Okay, so let's just think about, for example, whether or not an admission date is a quasi-identifier. I mean, lots of people are admitted to hospital every day, so you would think that hardly identifies anybody. Lots of people are, and they're admitted with all sorts of different conditions. But let us assume that we have a data set from, I don't have such a data set, but from Grenoble Hospital from 2013. And on a particular day, on December 29th, we have a record of someone with a severe traumatic brain injury or even severe trauma. Now, severe trauma is rare, but it makes the news. And so immediately we'll be able to identify with a background information attack from the newspapers, publicly available knowledge, we will be able to know that we have Michael Schumacher's data. Again, completely hypothetical, of course. So what I really want to get across to you is that anonymous is context specific. And so if data is shared to someone for one purpose, then it cannot be reshared with other people and then used for other purposes, because we don't then have an assurance of what the context is. Can we assess the anonymity? Well, we can. There are a number of mechanisms which allow us to actually formally calculate the degree of anonymity or re-identification risk. The one that's probably most famous or most well known, but there are other alternatives, is something called K-anonymity. And this really is an assessment of the degree of anonymity. And its definition is that there should be in any data set at least K records per any quasi-identifier equivalence class. So in any unique group of quasi-identifiers should never bring back less than K records. And if it does, then you can either suppress records or aggregate records in some way, or aggregate quasi-identifiers so that you always see at least K records. And if K is 2, then legally you are de-identified. It would always be a supposition as to who this person was, but typically we would want K to be a small number. And this prevents identity disclosure, although not attribute disclosure. It is vulnerable to a couple of different kinds of attack, though. The first is a homogeneity attack. So if all of those K records, or K plus whatever records, are all HIV positive, then unfortunately we have still disclosed someone's sensitive information to that quasi-identifier equivalence class. And the other kind of attack is obviously the background knowledge attack. I've shown you with the background knowledge of the newspapers, I could identify Michael Schumacher's data, but other organizations will have access to different kinds of degrees of background knowledge. And so in the data access committee, when you put in your request, or the HRA will be interested in who you are, what background knowledge you are likely to have, what linkage you are likely to do, and what homogeneity and K-anonymity of the data is. So these are considerations that need to go into specifying the data that you want from the electronic health record. There are some extensions to K-anonymity. The next, probably most well-known one is something called L-diversity. This is aimed to limit the background knowledge attack. So here says that in any quasi-identifier equivalence class, that there should be at least L distinct and hopefully well-represented sensitive values, so they're not all going to be HIV positive. In my example, this is actually really difficult and probably unnecessary to achieve, and doesn't acknowledge that there may be, just because two things are different doesn't mean that they're not semantically similar. So this is problematic. To get around that, there's this concept of Tor closeness, so that the sensitive attribute distribution within each equivalence class should be within a threshold, sort of an Earth mover distance, Tor of the whole data set. There are lots of ways of actually looking at this, but it is possible to actually put some numbers on how identifiable things are. We did this for the Amsterdam UMC data set, publicly available data set, and calculated for the friendly researcher, in other words, the researcher where we just want to make sure that they aren't able to identify the person sitting next to them. The rogue researcher who goes and looks in the newspapers and tries to identify the Michael Schumacher data set, not that it's in the Amsterdam data set, but who will have more background knowledge, and the rogue insurance company who may have access to social other demographics that could be used. And we're able to calculate a final risk of re-identification and put it to the public, and the public were happy. It's a little bit more, though, than just de-identification. There is this concept of disclosivity that we need to think about. So the sensitive attribute, as I said before, is the thing that we don't want to disclose. But disclosivity also depends upon the amount of embarrassment or harm that could happen from the disclosure of a sensitive attribute. So For example, this is a made up example, but I have had very similar cases come to the Data Access Committee where someone just wanted a diagnostic code. So just tell me what diagnosis these patients had. And that would be a very reasonable, it seems, thing to do if you're trying to find similarities or prediction for building prediction models. So you might get for a particular patient the following the diagnostic codes. These are ICD-10 codes. Bearing in mind, it's going to be very hard to go through all of these if there are hundreds of thousands of data records, and there may be, each patient may have many, many diagnoses. But if you do, you may find examples like this. So J45.9, asthma unspecified. Well, I think not particularly sensitive, unlikely to cause harm or embarrassment. E66.1, okay, well, this is perhaps slightly more sensitive thing. It's a gradation of things. Y07.1, other maltreatment by parent. Okay, we're now getting into rather more difficult territory. T74.2, sexual abuse, and then 004.8, medical termination complete complicated by pelvic infection. Now, suddenly, this combination of medical codes has said to a high degree of likelihood that someone has been sexually abused, probably by parent, resulting in a pregnancy that was terminated and ended up with a pelvic infection. And I think we would all agree that would be a completely unacceptable thing to have in a record and out in the public domain. So together with an approximate admission age and date, that might be really problematic. So we do a little bit, typically, to remove some restricted ICD-10 codes that would be particularly sensitive. But again, it's really context sensitive, context specific. So that's why when applying for data sets, people are a little reluctant just to give all diagnoses, because it's very difficult to know in combination exactly what one might, what embarrassment or harm might emerge from the knowledge that's inferred from that. Free or unstructured text is a particular concern. I should say it's a potentially very, very rich source of information. There's a huge amount of medical information in unstructured text, and it is ripe for exploitation and analysis by natural language processing or large language models and so on. And there could be potentially huge benefits in doing that. Free text occurs in two different places. There's free text sometimes in structured data, or should be structured data. This is generally a curation nightmare and is probably best avoided. But really here I'm talking about the free text blocks, such as hospital notes and letters and so forth. The issue is not that this is not fair game for research. It's just that anonymization is more difficult to actually guarantee. There are tools, Cogstack, and locally a tool called CRAID, but there's always an inherent residual risk, and none of this is binary. It's about the degree of risk and whether it's acceptable or not. So this kind of research is perfectly doable, but is likely to need some tighter controls on the degree of data access, where that data sits, and some kind of mechanism and agreement so that if the researcher detects that actually something was not de-identified, it can be fed back so we can try and correct the problem. But it's not an insurmountable problem. Just to finally talk about GDPR, the identification does mean that the data isn't subject to the GDPR, but the product, right, the final product will not necessarily use de-identified data. So just bear in mind that when you are using data that's de-identified, ultimately, if you want to translate into clinical practice, you will be using data that's real life world data from a hospital. And it's important to make sure, therefore, that these data are handled respectfully and isn't, for example, stored somewhere or computed somewhere, such as a large language model somewhere else in the cloud, that isn't in the EU and ideally in the UK. I'm going to quickly blitz through the ideas behind data quality cleaning and harmonization. Really important thing for me to sort of stress to you is that routine data, that's the data that you want to use for your models, was not collected by people who have your analysis in mind. Okay, it was collected as part of routine clinical care, and therefore the routine data has biases. It's got variances in definitions between different data sets. There are errors in there, and there is missingness, and it's not generally random missingness, it's often informative. And crucially, the reproducibility of your results and whether you will just get a fluke or not will be limited by the data curation that you do in the curation pipeline. And that means version control and thinking very hard about how you get from your raw data set into something more refined that you can put into the model. Now, hand data curation is time consuming and unbelievably painful. I've done hand curation for a number of big data medical projects, and it's something I hope to never have to do again. Of course, your manual deployment will ultimately, your deployment will never be able to rely on manual data curation, because your system, your final system, will just have to take the data that comes out of the electronic health record. Okay, so your AI system has to be capable of working with real world data. So there will be some data cleaning curation, but it needs to be a pipeline. And the data curation testing and sensitivity analysis needs to be an integral part of your development and redevelopment, because you cannot guarantee you will end up with the same results with different quality data. Here is a data. This is some calcium measurements from a number of different hospitals. And you would look at that and think, that's interesting. Calcium has a bimodal distribution. But I can tell you that calcium does not have a bimodal distribution. Calcium, what's happening here is there's conflation between two different kinds of calcium measurement, ionized calcium and total calcium. And because it hasn't been specified properly, we've ended up with, in different hospitals, we've ended up with what appears to be a bimodal distribution. You can imagine the impact of getting that wrong and moving on to a different data set in different hospitals. So really important to think about that curation pipeline. And here you can see the distributions from various different hospitals that actually they're choosing one or the other. I've published some data quality guidelines, which I just encourage you to have a look at. These are things to think, none of it's rocket science, but things to think about when you're designing your curation pipeline and your curation pathway. But you have to evaluate the data quality as part of your scientific evaluation. Otherwise, your model it's very hard to know whether your model really works or not. As part of that pipeline, it's best to try and use, and this is to try and avoid this calcium issue, standardized nomenclature. So very easy to take a data set from some area with different labels, build a model around it. But there's another step that would ideally one would use, which is to map those concepts to a standardized interoperable data format, so that we've got crucially semantic interoperability. The meaning of the data is well defined, so that when you take a different data set or try and deploy somewhere else, that you are not going to run into unexpected problems. There are lots of different kinds of vocabularies and ontologies out there in the data world. Many different ones, and it's best to probably try and use these rather than trying to bespoke things with individual hospitals, because what one hospital means by calcium may not be what another hospital means by calcium, etc. I may, on the interest of time, actually just skip over this, but just really to say that it would be great if all the hospitals just did the same thing and adopted a standard data model and terminology. And that's great for research, but typically that is never going to happen, because there are legacy systems, many different standards, and hospitals just will do what they want. So you need a layer in your pipeline that first takes whatever your raw data you're getting and maps it to something that actually is meaningful and standardized. Okay. A typical way of doing that is to adopt a common data model. So that is to say that we've got data set 1, data set 2, they're different, but we have a flexible master data dictionary model, and we, as best as possible, map onto that master data dictionary, so that when data set 3 comes along, we don't have to redo everything, we just map it again to the master dictionary. And there are a number of common data models out there. Probably the most well-known one and most utilized is something called OMOP. There are others. Again, I flag this as something for you to for awareness and to perhaps consider using in larger projects. So lastly, I want to think about software as a medical device. And I appreciate that I'm really concentrating on that left-hand side, that preclinical moving into phase 1 type study area. But it is worth just having an eye on the fact that ultimately, if we create something successful, any piece of software that processes medical data for medical purposes to change management of a patient is a medical device. Okay, and there are laws, and that doesn't matter. So an Excel spreadsheet programmed with a risk calculator is a medical device, and there are laws defining how these can be used on people outside of consented research anyway. Okay, and so typically, once you have a device, these devices need to be CE marked or the UK equivalent as a medical device, and then we know, And then what the CE marking does, it doesn't actually say that this is an effective treatment or an effective device, but it says that it's been appropriately tested and we know how it is going to perform and who it is meant to perform on. There are regulations. These are the European regulations, which are essentially mirrored in UK law surrounding medical devices generally. These are obviously not bits of software, but in the European Union, we classify medical devices into four categories: low to high risk. So low risk would be this sling, this bandage. Medium risk would be this intravenous drip because it's invasive but not going to be there very long. Medium to high risk, something delivering radiation, and high risk, something this is an extracorporeal circuit. Now, it turns out for class 1 medical device with low risk, I think this is good news. Because actually you can self-certify your CE marking with that. You don't need to go through any process at all. Unfortunately, if we look at Medical Device Coordination Group document, it is extremely unlikely that anything that actually influences a patient is going to be class 1. So everything that you are working on as prediction tools, if deployed, would be at least a class 2A and up, or even up to class 3, if it potentially could lead to severe harm or death if it went wrong. So we are going to have to respect the medical device laws. Now, there are ways around this. Article 5, I'll spare you the task of reading all of the EU regulations. Article 5, Section 5, so Article 5.5, which is this bit, does allow for an individual institution to deploy a medical device locally, provided that it doesn't go anywhere else, it's only deployed locally. So if you came up with a great thing and you thought that we want to deploy this in Addenbrooke's hospital and actually see how it works, we can do that. We don't need to go through full CE marking, but what we will need to do is use a software quality management system. So we need to have some processes that we can document that this has been tested properly and we know how this is going to work. So that's Article 5.5. There are a number of international standards and related documents. The ones that you may want to know about if you're interested in it, particularly interested in this later domain, is the ISO standard 13485. This is the standard for quality management system. This is what you need to do in terms of paperwork to actually make sure that you are following a process which is reproducible for building a medical device, software medical device. And related documents that are useful to view in combination with this is the IEC 62304, which is medical device software lifecycle process, and 82304, which is a little bit less relevant. It's about actually how to make a product, which I'm really not going to talk about here. But essentially, this is the guts of it. And I'm just going to talk through this in a little bit more detail. This is the software lifecycle management for software development. And there are similar life cycles for deployment and monitoring. But the crucial thing is, if I can draw your attention, that we start with this rather quaintly state of things. What are the customer needs? And that feeds into this idea of software development planning. Now, let me give you an example of what I mean by this and why this is important. I recently was involved with a particular risk scoring software, which was being used. It had been implemented, and because of a rounding error was producing, it turned out was producing slightly different results to the web implementation of this particular software. So it took some risk factors and predicted whether or not a patient was likely to have sepsis and could be treated for that. Now, this is not a CE marked piece of software. In fact, the risk was very, very low. That rounding error wasn't probably clinically significant, but is unfortunate. And does demonstrate that it hadn't been tested properly. But when I went and spoke to the end users, it turned out that this piece of this risk model, which had been very, very carefully developed for a particular group community, for a particular group of people, was in fact being used as part of another stratification. So there'd be a first stratification, pre-stratification of patients, and then the risk model being applied. Now, that is not what was conceived when the software was developed. The software was developed for the purposes of someone risk stratifying the whole cohort, not just individual, a segmented cohort that's been pre-stratified. And you will all know that the performance of a risk model on a pre-stratified or a smaller group of patients, on a different group of patients with a different risk profile, will not behave the same way. It may behave extremely unpredictably. And so this is really where actually defining what is the software going to do. It's not just is it going to predict, but is it going to, what is it going to predict, in who, for what purpose, because that purpose will define what the performance of the software needs to be. So for example, if the purpose is for screening, then that will mean that a risk model will have to have good would have to have good sensitivity, but doesn't necessarily have to have quite such good specificity. Whereas something is diagnostic, to then do something high risk in a different context may require a different trade-off between sensitivity and specificity. They're really important to define right at the outset, what is the question that this piece of software, this model is actually trying to address, and in whom is it going to be deployed, and how is it going to be deployed? And that's this kind of software development planning and that requirements analysis and architectural design. Then moving through, there's a whole testing piece, which is, and that requires testing of not just the final piece of software, but what about all of the libraries and packages within that? How robust do we know that a piece of software is to a change in Python library? Now, you, of course, as software developers, in this context, software developers can't be responsible for the next Python library, but you will probably should be thinking about exactly what Python library version have you tested it on, so that we know that if a Python library changes, then actually we need to go back and think about retesting this, or alternatively locking to a particular library. So those are all the considerations that are really important if we actually want to get a piece of an AI model into clinical practice. So I'm going to stop there. So hopefully some time for questions. But just to say, in order to make a difference, we really, really want to encourage translation from the bench to the bedside. And to do that, we have to use real world data. We really need to get our hands dirty and use real world data. Now, it is true that medical research is very highly regulated, okay? But the regulatory framework for de-identified data is genuinely really very easily navigated. You will need to buddy up with a hospital, and you'll need to treat the data with respect. And if you don't, there's a chance of getting into a lot of trouble. But it's not difficult to do this. I would encourage you, as much as it makes sense, to build with some kind of deployment, at least in mind, within reason, because you are working in the domain of making software as medical devices. So think about your data curations to a great extent as possible. Use standards. It helps you reuse your software as well, and consider the software lifecycle. And I promise you, I work with really very sensitive data, real world data, de-identified and not de-identified, all of the time. This, I promise you, is not hard. There are huge opportunities out there for you, but you need to buddy up with someone that knows what they're doing. Thank you very much. Very happy to take some questions if there are any.
Great, thank you very much for your talk. So there's a bunch of questions that have posed through the talk. So Angus, if would you like to unmute and ask your question?
Yeah. First of all, that was very great presentation. I'm just curious about if the process for accessing the data biomedically would be the same in the US, or it would be different, because one of the things I'm interested in is trying to, as it's well known fact that the US aren't very good. And my topic is trying to identify which the states that have the worst health, which state has the best healthcare, which one has the worst, then show a contrast and highlight how using machine learning or AI can identify and improve it and such. And one of the things I'm concerned about is the ethical aspect of it, of like collecting the data. And I'm kind of unsure of how to go about collecting the data and such. In that regard, that's why I'm asking, like, are there particular policies or something that are
So the regulations in the US are very different. Well, I say they're very different. On the plus side, broadly speaking, the regulations are more permissive than in the EU, because there is less of a true description of what de-identified means. So that actually makes things rather easier. I think the question for me, though, really that springs to mind is in the detail: is where would you be collecting these data from? Because what you're suggesting is a multi-center, well, a complete national data set, not one that's controlled by any individual hospital. So if for things that are controlled by particular hospitals, then the slight complication in the US is that the ethical approvals are local. That makes them a little bit more agile than the UK version, which is for the whole nation. But it does mean that you have to go to each individual data controller individually and get each individual site to get the ethical institutional review board approval, IRB approval. But I think it really depends on where you're hoping to get this data from, because if it's from census data or survey data, then probably all of that will have been dealt with before.
So my intention is to I look doing research. It says the best place for healthcare or medical insurance in the US. The best one is I believe the Hawaii, and the worst ranked one is Mississippi. So would I need to try and do research on the main hospitals in those states and access their data or request their data that way?
So one way of yes, that would be one way of doing things. But it'll be hard because it's just logistically going to be tricky. Another way of doing, or perhaps getting a similar handle on things, will be to look at Medicare, Medicaid type data in the US and claims data, which is another rich source of data, slightly different sets of biases in. Another possibility, but it's a little bit tricky because, of course, the healthcare system in America is quite stratified. So by picking a particular healthcare system, then you obviously bias the people that you're looking at, and you're immediately biasing to those people who have. This is the one difficulty. If you're looking at hospital data, you are immediately biasing to those people who access the hospital. And unfortunately, that means you're missing the people. If you're looking at healthcare inequalities, you're missing those that were unable to in the US.
So it's like trying to say, I'm trying to highlight, how medication access is difficult. It can be difficult for certain classes of work in the US. So, say the working class don't have the money or insurance to access the medication they need, but somebody who's much richer does have access and can access it easily. I think what I'm trying to do is trying to avoid biasness as much as I can. Just trying to highlight, it's not the working class that suffer, but also the middle class and the upper class, if I can find data relevant to that.
Yeah, and again, it's just a little bit difficult in the US because they will tend to use different healthcare systems. There are some US-wide healthcare systems for Kaiser Permanente, Veterans Association, and so on. So there are some organizations that may be able to look at hospital, practice across a wide areas of the US, but it is difficult. The US healthcare system is very, very fragmented, unfortunately. That makes this very difficult.
Okay. Yeah, I think that's all the questions.
Thank you.
Thank you.
Okay, so Nick, if you're ready, please unmute and share your question. Well, I can ask it myself otherwise. I believe he was interested on government-funded initiatives to support academic spin-off companies with software as a medical device development.
I'm sorry, say that again. I just missed
if there's any opportunities for government-funded initiatives for spin-off companies with.
Yep, absolutely. There are. So the government does have a number of. So Innovate UK, there are a number of, and the NIHR, which is essentially the NHS's research wing, it has a reasonable amount of money actually, are very interested in bench to bedside work. So things that will translate into the near future. So absolutely, there are government, Innovate UK being the really obvious one, opportunities for exactly this early kind of early industrial kind of spin-off. Absolutely, the government's very much trying to promote that.
Sorry, now we can actually speak. Can I ask this? So I'm working with a company that have got a lot of that funding, but the difficulty is then moving into quality management systems. Even the concept of having requirement specification is something completely new. So like there almost needs to be a centralized system that's like a hand-holding thing that offers advice and expertise to like do the job, so that these small startup companies don't need to become experts in all these different fields because they don't have the funding to get people to do those roles.
Yep. I totally agree with you. I think this is an area that has received, and in many ways, one of the reasons I wanted to bring it up in this talk is precisely because it's an area that hasn't received a lot of attention. What I would say is that many universities do have outreach spin-off type functions which are able to assess. Certainly my university does. In fact, even my hospital has internally within clinical engineering ISO 13485 accreditation and a service to provide this. It is not, I should say, it's not free because it does require quite a lot. If you're going to produce a complete, proper, safety file, technical file on a big product, it's not a small thing to do. It does require some money, but there are people out there, often in academia, that can help, and sometimes in hospitals. But I agree with this. It's not an easy area, and it can be impenetrable.
Thank you.
Okay, so the last questions maybe before time runs off. Ahmar, if there's any outstanding doubts after Samuel's answers.
I was just looking at there was one. I was just saying I was just going to flag up. There was one question in the chat talking about routinely collected data anonymized just need Caldecott approval. That's not correct. You do require, if you're going to do research on it, you do require an HRA approval, or go through some mechanism like we have in Cambridge for ARIN.
So can I ask like a related question on this? Okay, great, thanks. So yeah, I mean, you talked about accessing these de-identified data routinely collected. I mean, this is great, but I have an opinion and I want to know what you think. I think this alone, like if we access these routinely collected data, that's not going to be good enough because these are all historical data. You need to do these real world studies where a clinician can see the outcome of an AI and then they can manage patient and see how that influences. And I think this is a big barrier going from those historical data to prospective study. And I just wonder, I mean, this probably, like what you said is great. We need to access this routinely collected data, but I think this will not move the field forward unless we can ensure that we can have these prospective studies, which I think there was a scoping review recently. Over 10,000 studies, only 8 studies were prospective studies. So we have a huge problem here.
You're absolutely right. And that's really where if I go back to the first slides or the early slides I put up, you have to go from some point from the preclinical, right, which is your retrospective, whatever, or shadow deployment prospectively into phase 1, phase 2, phase 3 kind of model. All right. And crucially, I should just say, it isn't just the AI model, it's the AI model in the context it's deployed. Okay. So just to give you a really trivial example, if you produce a prediction model and I say, okay, well, how will you deploy this? Well, I want a pop-up that says, actually, this is a high-risk patient. Guarantee you that it won't work, right? Because clinicians are notorious. I have hundreds of thousands of pop-ups that I'm suppressing. It makes no difference. People ignore them. So it's how is you going to embed that into the actual clinical workflow? And that's where you get into your phase 1. Is it actually safe in clinical practice, phase 1 study? Now, bridging that gap, you're right. At some point, you need to go into prospective mode. But if you've done your software engineering properly on your retrospective data, then you can come into hospital and do that prospective. Now, not all hospitals have the facility to do that. But just to give you an idea, my PhD student, a recent PhD student, has developed a nice time series model for the intensive care unit. And we're going to retrain it now in the hospital, on hospitals. So I have cloud compute within the hospital within the hospital. We will retrain in on the hospital data set and then run in shadow mode prospectively to evaluate the prospective performance. And then we can move into the phase one part, which is actually how do you deploy it back into the electronic health record. Those bits will require deep integration with the hospital. There's no way around that. But even before that, you need to use the retrospective data to act and do it properly to make sure that you're going to be compatible and it's going to work at all. So I think it's a stepwise thing. Yeah?
Thank you. This is really helpful. And one additional point, question at this time: where do you see the value of? I know the value of randomized control trial, because I've been in meetings where people talk about that. Most of RCTs don't get implemented. But how do you see RCT and then AI algorithm? Do you think it has a place or it doesn't?
Yeah. So do you mean an RCTs of the AI algorithm?
Yes, having a study design that's RCT.
Yeah. I think so. I think the difficulty with anything is that nobody believes. So setting aside the fact that RCTs, that they may be a gold standard, but they don't actually prove causality. They're a good, probably the best we've got. No one, unfortunately, believes you unless you've done some sort of randomized control study. Okay. The question is, what randomized control study do you want to do? Because if you want to demonstrate the AI algorithm improves something a long way down the line, like survival, that's really hard because there are lots of other things that can get in the way and mess things up and mean that you don't see the signal down the end. So actually, it's think about AI process measures of process RCT. So, does it improve the time to referral? Does it improve the time to some other action? These process measures, absolutely. And yes, ultimately, in order to get things adopted, you need some sort of RCT. Otherwise, it just doesn't. It's difficult for the community to really accept that. And I can guarantee that if you don't do an RCT and get the other way around it is to get the product CE marked and introduce it. But someone will come and do an RCT at some point in the future, and those have sometimes turned out to be embarrassing when it's turned out that the product, I think medical products rather than software, have actually turned out not to provide the promise that they claim to provide.
Thanks a lot.
You're welcome.
Great. So I believe our next speaker is here, so we'll have to close here. Thank you very much for your talk.
Thank you. Thanks. Nice to see everyone. Goodbye. Bye.
Thank you so much. And I think we're going to take over from here and move to the next causality related talk, but we're going to talk maybe about how RCTs are not the only thing out there that we can rely on. As you probably know by now, my name is Juliana, and I hope you'll remember me as the first year PhD student who kind of works in causality. I would like to introduce to you today our next speaker who will further dig into the world of causality, Dr. Alexis Bellot. Alexis is a graduate of the Van der Schaar Lab who finished his PhD in 2021. After his PhD, Alexis completed a postdoc in causal inference at Columbia University and then joined Google DeepMind in London as a research scientist, investigating causality and its application. His goal overall is to improve decision-making with data in applications such as healthcare and to guarantee the safety and alignment of AI algorithms. Today, Alexis will introduce you to the concepts of Pearl's causal hierarchy and distinguish between the observational, interventional, and counterfactual statements that we can make about the interactions between variables in complex real-world systems. He will also explain how the causal inference framework relates to other machine learning paradigms, such as transfer learning or reinforcement learning. Before I pass the virtual microphone to our speaker, I would like to remind you all and encourage you to post any questions which may arise in the chat throughout the talk, as we will then try to answer as many of them as possible during the Q&A. Without further ado, Alexis, the floor is yours.
Thank you very much, Juliana. I'll share my screen if that's okay. Right, so you should be seeing my screen if everything worked correctly. So it's very nice to be here. Thank you very much for the invitation and the kind introduction. As Juliana said, I want to give you a little bit of an introduction to causality as a field and to the problem of causal inference. I'll try to keep it on the lighter touch right? Give you a little bit my perspective, some aspirations for the field, and all the different kinds of applications that fall under a causal inference task. If you'd like to follow along with your own set of slides, I've linked them to in this link. So feel free to go there, download the PDF, and follow along. So I want to start maybe by giving you a little bit of my own motivation for joining, for studying causality and causality from data. Which is mostly that I see causality as a root node problem that as humans we've used to define a lot of the intuitive notions that we have in several different tasks and how we go about our own day-to-day tasks. So these involve concepts of blame and responsibility, agency, interpretability, ideas related to robustness, decision-making, transfer learning. All of these in some sense can be defined from a causal perspective. And so solving problems in causality has potentially all of these different ramifications to help our understanding in all of these different fields. So it might be, so from this perspective, it might be easy to understand that causality has appeared in many different fields across time, including statistics, computer science, but also psychology, philosophy. And in my own reading, there's really two very distinct approaches to understanding causal inference problems. One of it you might think of as the logical approach that appears most prominently in the computer science and statistics literatures, where the task is to automate the derivation of causal conclusions from typically very well articulated assumptions. Right? So I'll talk about this much more in a second, but you can contrast this logical approach with one that is much more human-centered, that instead aims to generate causal conclusions in a much more human-like way that typically relies on our own intuitions of causal and subjective judgments, and that relates much more directly to the rich human experience. So you typically go beyond co-occurrences of events in data and take into account temporal information, spatial information, language, culture, all the ways that humans have to learn about the world. And that is typically also intertwined with learning everything else, right? And these are two different research programs that you can embark on to learn about causality. And they've typically been quite distinct. But I want to mention this trade-off now because with the advent of foundation models that are trained on language, images, video, the way of understanding causality in machine learning is kind of shifting a little bit in my mind from the logical approach to the human-centered approach, because these language models plausibly understand causality in a much more of a human way than from a logical approach. So it's worth keeping in mind this more expansive view, even though in statistics or machine learning or typically more the data sciences, a logical approach is much more prominent. So from a logical perspective, to try to formalize causality and causal inference problems, suppose that you're studying a system and you get to measure or observe some of its attributes. So these are typically variables, some determinables of your causal setup that take a range of values, right? And that are interconnected in some complex way. Now, when I draw a picture like this one, a graph, I really aim to think of this as a summary of or a representation of an underlying structural causal model. So here, a graph is just meant to summarize the functions that relate one variable to another. So for instance, when I draw an edge between two variables x and y, I mean that there's a function that takes as input a function for y that takes as input x, as well as potentially additional exogenous influences that impact your system, but you don't get to measure, to produce a particular value of y. So you can think of these underlying models as just a collection of functions and a collection of unobserved variables that you capture the uncertainty around them with a probability distribution. So a graph is nothing else than a summary of this underlying system. And the first important observation is that if you take measurements from this system, if you sample from the structural causal model, you get some evidence about the probability of events when they're caused in the normal way, right? So these are some evidence for the probability with this P notation for different events, different values of the measured variables in your system. Now, often you might want to reason about interventions of your system, actions, manipulations that would explicitly modify some of the mechanisms in your underlying setup, fix their values, and then hopefully predict the behavior of the rest of your system under these interventions. So in this setup, what's happening is that you're explicitly replacing some of the mechanisms of your system with some fixed value that you as an experimenter have chosen to fix, right? So this potentially completely changes the dynamics of the co-occurrence of different events in your system because you've modified some of the underlying mechanisms. And so probabilities under intervention, right, these so-called interventional probabilities, are really qualitatively different and don't necessarily have any relationship with your observational probabilities that you would get by just passively observing your system, right? So in the literature, there's different notations that people use to express interventional probabilities. Maybe the clearest one is the one that involves this M4 model, where you clearly see that it's the probability of the model under this intervention on, say, a variable x. Now, just for completeness, though, I'll talk mostly about interventions in this talk. You can also reason about more sophisticated statements about the world, right? And these are so-called counterfactual probabilities. And they typically mix statements about two different hypothetical scenarios, right? So maybe questions like your patient has had a headache for a number of days, and you wonder about the probability of that persistent headache to take place had you instead given it a treatment a week ago, right? So this is a totally plausible statement to make and reason about, but they make a contrast between two different worlds. One in which your patient hasn't taken the treatment and has this headache, and one in which you went back in time, if you want, and you're imagining this hypothetical world where your patient did take the treatment and want to make some inference about the likelihood of him having a headache at this point in time. So these are called counterfactuals and are typically expressed with this kind of notation where an outcome can be thought of as occurring after some treatment or without the treatment. And what's interesting about counterfactual probabilities is that really there's no experiment that you can imagine that would give you direct evidence of these probabilities, right? There's no way that for the same patient you can both give the treatment and not give the treatment. So in some sense, they're not empirical, right? But you may still wonder about them. There's many different questions that are relevant from a counterfactual perspective. So the second one that I'm showing here, where you have this combination of the outcome on the two different interventions, is another one that's very commonly used, also called the probability of sufficiency and necessity. And this one, for instance, you're looking to reason for, imagine you're a resource-constrained hospital that only wants to give the treatment to those patients that would, in fact, respond to the treatment, and simultaneously those patients that, if you didn't give the treatment, would have a bad health outcome, let's say, right? So you don't want to waste any resources on patients that would have improved if you didn't give them the treatment. And these kind of statements is captured in this type of counterfactual probabilities, for instance. Now, the reason why causal inference, so trying to automate, trying to derive, generate conclusions to all of these different answers from data, the reason why that's so difficult is because observational data almost never uniquely determines the effect of interventions. So in a way, even if you were able to make infinite observations of your system, that still vastly underdetermines the value of interventions. So absent further assumptions, there's no way for you to predict the effect of interventions just from observational data in, let's say, a purely data-driven way. So this has been formalized in several different mathematically precise senses, and to some extent is an intuition we've had as humans for a very long time. But now we really know how challenging this problem is. And so if you like this model-based perspective to think about causality, what I'm really saying here is that you start with a space of all possible causal models, right? You make some observations of your system, which constrains the set of all possible models for your system to a smaller subset, here in light blue, right? And this statement that you're never able to uniquely determine the effect of interventions really means that if you were to take two different models that are both compatible with the observational data, you would most likely get two different answers for the causal effects of some outcome under some intervention for these two different models. Okay, so is there anything that can be done? Because I guess there's two different ways. So once you realize this challenge, there's two different ways you could go. Either you give up and think of interventional and counterfactual quantities as non-empirical to some sense, and statements that we shouldn't be talking about if we're hardcore data scientists. And to some extent, this has been true. This has been the predominant approach through a lot of the 20th century, and we still have that legacy in a lot of the medical studies, for instance. But simultaneously, there's also another approach you could take, which is to explore different types of assumptions that would allow you to make progress and answer these causal questions uniquely, right? And so let's say the first attempt at formulating additional assumptions and try to nevertheless come to causal conclusions is due to Sewell Wright in the 1920s. So he was interested in understanding the transmission of heredity to the transmission of, let's say, the fur color in a guinea pig and the influence of heredity and the environment on the color of these animals, right? And he came up with this qualitative model, right? That the picture you see here that relates different variables together and assigns, in this case, certain coefficients to each one of these edges. So this is his own understanding of how transmission from one generation occurs in a, let's say, coarse qualitative sense, right? And he was able to relate the correlations in data with the coefficients that he imposed on this model to get a unique prediction for the effect of intervention. And if you like, this is one of the first attempts from a more quantitative perspective to combine some knowledge of the system, some assumptions on the causal structure, combine it with data, and get some verifiable causal conclusion that holds if the model is correct. And so since Sewell Wright, there's actually, and if you take this model approach, a way to understand what he did is really, with his additional model, he was able to further constrain the space of compatible models with its data and its assumptions. And this further restrictions then generated a smaller subset of models that he had to consider. And in his case, any two models compatible with his assumptions always gave the same effective intervention, the same causal effect, right? So this is a little bit from a model-based perspective how you can understand, let's say, identifiability of causal conclusions given data and assumptions. And so starting in the 1970s and 1980s, there's this research program that emerged following the footsteps from Sewell Wright,